For the introduction, and hi everyone. Thanks for being here. Thanks for having me at RIKEN-AIP. It's always nice to be here. I'm Australian, so good luck with my accent. I went for a long, complicated title, and hopefully it becomes clearer over the next 45 minutes or so. This is joint work with these fantastic people. So Jarno Alanko, who's a postdoc in our group at the University of Helsinki. Keep an eye on this guy. I think he's just getting warmed up. Elena Biaggi, who's a really brilliant doctoral researcher in our group. A lot of what I'll talk about will go into her PhD thesis. Joel Mackenzie from the University of Queensland was visiting us about a year ago. We got some of this work done. Joel has background in search engines for web search engines. And I'm trying to mislead him into genomic search engines. So far it's working. And finally, Jakub Wortaniemi, who did his master's thesis in our group a little while ago and now lives in Japan, and I believe is soon to be starting here as an intern. So these guys are really the driving force behind what I'll talk about today. All right, I'll try and give you an idea of the problem, which is indexing k-spectra. So the k-spectra for a string X is the set Sk of all the k-length substrings that appear in X. And I'll call these k-length substrings k-mers. I think this is terminology that's familiar to most of you. So here's an example I prepared earlier. This is X, T, A, G, C, etc. And this is its k-spectrum. So these are the 3-mers that appear. So k is equal to 3. These are the 3-mers that appear in X. And if you're very observant, you'll notice that they're in co-lexicographic order. So I grew up thinking about things in terms of lexicographic order, but I've been sort of dragged into co-lexicographic land. So by co-lex order, I just mean if you reversed all of these k-mers, they'd be in lexicographic order. Okay, but for various technical reasons, we'll have them in co-lex order throughout the talk. This will be important. All right, and so what we want is to pre-process the spectrum and build a data structure that helps us to answer membership queries, k-mer membership queries. And k-mer membership query, which I'll call lookup, just takes a query k-mer, Q, and it returns the co-lexicographic rank of the k-mer in the spectrum, if it appears in the spectrum. And if it doesn't occur in the spectrum, then it returns some sentinel value, so 0, I've said here. So lookup query for ACA would can't see my pointer, would give us 2 because ACA has colex rank. It's the second k-mer there in colexicographic order. And lookup for AGA would appear 4 because that's where AGA is in colexicographic order. And lookup for TTT would return 0 because TTT is not in the spectrum. Okay, so this is the problem we're going to be focusing on today. Giulio's here. Nice of you to join. All right, and a couple of important things. So k is a lot less than the length of the input string. Okay. Typically, we're talking about k in the range 20 to 100, something like that. So just keep that in mind. And also, the alphabet size is small. So we're interested in solving this in relationship to genomics problems. So are we sort of assuming a DNA alphabet? This problem might also be interesting on general alphabets. But this is where we'll be today. So this is like basic computer science stuff, right? Very simple, very sort of clean CS problem, algorithms, data structures problem. And so there's been a lot of prior work on it. 2 main strands. So the first strand is the BOSS data structure, which is named for its authors, Bo Onodera, Sarakane, and Shibuya. And it was discovered nearby over at the National Institute for Informatics more than 10 years ago now. And it's been implemented in a few bioinformatics tools, and it has a reputation. It's very small, okay, but it has, Sada's not here, so I can say this, has a reputation of being very complicated to implement and slow in practice. So that's one sort of way to solve k-mer membership queries. The other strand of work involves hashing, and this is kind of the first thought you have when presented with that problem. Like, oh yeah, I could store them in a hash table and look them up that way. And there are some very clever hashing techniques that have been developed for k-mer lookup. A few of them are listed here. And probably there's been more work on the hashing side of things so far. Hashing's winning, I think. And I'll talk a bit more about some of these methods later, very briefly. But yeah, so this is the state of the art. And really where we started this project was to try to understand deeply what was going on here. So what were they really trying to achieve when they set out to design this succinct BOSS data structure? And so we pulled it apart, and what we came up with, and is what I'll focus on in the first part of the talk, is the Spectral Burrows-Wheeler transform. What we found was that the BOSS data structure is just a particular implementation of a more general scheme. This Spectral Burrows-Wheeler transform, SBWT, I'll call it. And the SBWT, and we'll really get into the details in this talk, I sort of want to get into it in some detail so that everyone understands what the SBWT is, hopefully, by the end of the talk. But at a very high level, the SBWT takes the k-spectrum, which is a sequence of k-mers, or set of k-mers, and it transforms it into a set of subsets, okay, of the alphabet. Okay, and then by pre-processing these subsets to support particular types of queries, we can do k-mer lookups. So really, the Spectral Burrows-Wheeler transform transforms the problem of k-mer lookups into another problem called subset rank queries. Okay, and for subset rank queries, we have a very big design space. We can solve it in lots of different ways. We came up with a few of them. I'll talk about one of them today. There are some others, and there are probably many more. So, like, I encourage people to think about how to solve subset rank queries efficiently. This is something we published a couple of years ago, but it's like something that we've kept working on various ways. But there are open problems, and I'll talk a lot about them today. Okay, the second part of what I'll talk about is called batched k-mer lookups. So often in data structure design, we're faced with a problem. We want to design a data structure to solve certain queries, and we're assuming there's one query. We sort of come up with an algorithm to use the data structure to solve one query. And in the case of k-mer lookups, that's this lookup query. And when we have lots of different queries, we just call that lookup function many times. So if we have m queries, we call lookup q m times. That's our solution to the multiple queries. But in many settings, we get these large batches of queries. Okay, we don't just get single queries and then sometime later we get another one. We get a whole batch of queries arriving at once. This is the usual case, actually, in genomics. And so the question we asked ourselves, and what I'll talk about later in the talk, is can we support this kind of lookup batch query that takes all the queries as input and somehow solves the lookup problem m times faster than calling this single lookup query m times? Okay, and of course the answer is yes. I'm here to please. And we can do that using the Spectral Burrows-Wheeler transform. And this is also ongoing work, but we published some first results at LNX earlier this year. I think there are open problems here too, and things for us to think about in data structure, succinct data structure design in general. All right, so a little bit of motivation before I get into the details of the SBWT. So I've mentioned bioinformatics a few times already. K-mers are used in lots of different pipelines. Okay, these k-mer lookup queries. So it's a basic operation in these tasks that I've listed here and many other tasks. And so improvements to k-mer lookup are important. They have wide-reaching impact. And of course, as an algorithms person, a k-mer lookup, an indexing k-spectra has a certain amount of inherent interest to me. It's a nice, clean data structures problem. But some colleagues of mine at the University of Oslo, Jukka Koranda and Tommy Mucklin, they've actually been using k-mer lookups to solve real problems for a problem actually called bacterial lineage identification. So I thought it'd be interesting to sort of talk about this as motivation. The setting that we have is that we have a plate, like a laboratory plate, with some bacteria growing on it. And this might come from a patient, for instance. And we want to identify the strains of bacteria that are growing on the plate using sequencing, using high-throughput sequencing, maybe to assist in diagnosis or treatment. Okay, so the way this is currently done, or typically done, is that a lab technician will look at the plate, look at the colonies that are growing on the plate. Okay, so the plates might look something like this. And they'll manually pick the colonies that they see growing there, put them on new plates. Okay, and this can take time and effort, and it's error-prone. But okay, so if they manage to do that, it's like maybe there's some contamination that could happen. But they do this, and then they grow the plates up for a while, and they sequence what's on the plates. Okay, and then they would say, okay, well, these are the bacteria that we've found in these colonies, and that was what was in the original sample. Okay, and they'd make some conclusions based on that. And what Jukka and Tommy and their collaborators are proposing is that we would instead just sweep this plate, sequence all the material on it, okay, whether it looks like a bacterial colony or not. And then through some computational process, we'd process all these reads and determine what was actually there. Okay, so, and maybe we'd also determine the relative abundance of each of these strains. Okay, and then we could go and do diagnosis and treatment or whatever. So they've developed this pipeline that they call MGEMS, and it's being used and being further developed by them. But this is how I got involved in k-mer lookup problems. So that's why I'm mentioning it. And the first and the sort of most computationally demanding step in their process is as follows. So we start with this sequencing of the whole plate. So we've got these fragments of DNA, reads, okay. And down here we have a database of bacterial genomes. So it's worth sort of considering the numbers that are involved. So these are bacterial genomes. They might be about 5 million letters each, typically. And the database has maybe tens of thousands or even hundreds of thousands these days bacteria in it. So it's a sizable data set, okay, approaching hundreds of gigabytes or even terabytes of data. And what we'd like to do is take these fragments and align them to the genomes and find the genomes that they best align to. But doing proper alignment, like local alignment, is too expensive. Okay, so we do something called pseudo-alignment, which is, well, I'll tell you what pseudo-alignment is. So we start by building a k-mer index for our data set. So this is much smaller than the original data. Okay. Maybe 10, 100 times smaller than the database. And then we take a read. Reads are maybe 100 letters long, 200 letters long. And we slide our k-mer window across the read. So we take all the k-mers in the read. This is the first k-mer. And we look it up in our k-mer index. And we get a hit. And the k-mer index has satellite data which says which genomes the k-mer appears in. Okay, it appears in genome 1, genome 2, genome 898. And representing this color information, so this genomes that each k-mer occurs in, this is also an interesting problem, and I think maybe is the subject for the next talk by Giulio. That's good, getting a nod over there. Yeah, so we determine which k-mers, which genomes the k-mer appears in, and then we look at the next k-mer, maybe it doesn't occur. Look at the one after that, maybe it does occur only in these two genomes. And we do this for all the k-mers in the query. And then we come up with we apply some scoring function. This is another sort of interesting piece of research. And we try to decide which strains the read belongs to. Okay, and we do this overall, and we come up with some kind of abundance. Okay, but this is a heavy computational problem. And I want to emphasize that this is sort of something that we want to do fast. The speed is important, and it's not just because there's large data volumes and the data down here is sort of sizable. It's because bioinformaticians are always pushing the limits of these things. If you provide them with an index that's fast, they'll come up with a way to use it more. And to squeeze more out of it. And this is because bioinformatics and biology in general is often exploratory. Right? So these indexes should always be faster than they are currently. Efficiency is really important down here. Okay, so we have an index to do this pseudo alignment. It's called Themisto, and it's being used by Giulio and Tommy and a few other groups that we're aware of, and I just wanted to quickly mention that it's out there. But so that's enough about motivation. I'll get back to talking about this data structural problem from now. Okay, so this is the problem, indexing k-spectra. Hopefully it's still fresh in your mind. And the solution to the lookup problem, so not to the color representation, but to the lookup side of things, that I'll discuss is this spectral Burrows-Wheeler transform. And we called it the spectral Burrows-Wheeler transform because it sounds cool, okay? And also because it's essentially the Burrows-Wheeler transform of the k-mer spectrum, as hopefully I'll convince you. All right, so remember, we take the k spectrum as input, and we're going to produce this sequence of subsets. That's the spectral Burrows-Wheeler transform. All right, so let's define the SBWT to begin with. So here's an example string at the top. These are all the k-mers that appear in the string, and they're in colex order. Okay. So these are distinct 3-mers, sorry, not k-mers. So k equals 3 in this example. Okay, and I'll duplicate them here. Okay, and what we're going to do is draw edges between k-mers or 3-mers that have an overlap of length 2. Okay, so what am I talking about? So we draw an edge between $$$ and $$t because those two k-mers overlap by 2 characters. Okay. And it's useful to think about there being a label on this edge. Okay, so in particular the label on this edge is a t because that's what we need to add to the first k-mer to get to the second k-mer after we remove the first letter. Okay. So these edge labels are going to be important. This is the second edge, so we've got an edge going from CAA to AAG, okay, because there's that overlap of 2, and the label on that edge is a G. Okay, and this one goes to 4 different places. So that first arrow would have an A on it, and then a C, G, T. All right, here are all the edges. This took ages to make this slide, but there it is. So let's now clean it up a bit. And I've drawn it like this as a kind of bipartite graph. That's just for clarity. Yes, Giulio?
You only draw a line if the former appears in the string. Because you could have $$, $$, $$.
Yeah, that's true. Yeah, $$, is special. Yeah.
So it's special. So it doesn't have to. The former doesn't have to occur in this.
Yeah. Yeah, good question. So, and also, I just wanted, so I've drawn it like this just for clarity, sort of this bipartite graph, but really these edges are kind of pointing into the same column. Okay, it's just does my head in if I draw it like that. So I did a little bit. Okay, now we're going to tidy the graph up a bit. So what we're going to do is whenever we have a k-mer that has the same k-1 length suffix as the previous k-mer in lex order, we're going to remove the edges coming from that. So what do I mean? Hang on, have I skipped too far? Can you see the grey? Can't really see it. Can you? See that colour change? No. So over here we've got this k-mer has the same k-1 length suffix as this one. Here this one has the same. I've actually shaded them in grey, but it's not showing up on the projection. So these ones have the same k-1 length suffix. These three all share a k-1 length suffix. So I'm just going to remove the edges from all but the first of those groups. Okay. So that gives us a graph like this. So there's no outgoing edges from this one or this one anymore, but there are from this one. All right, and when we clean up the graph like this, we have a very useful property, and that is that now there's only one incoming edge to every k-mer on the right. There's exactly one incoming edge, except for $, $, $, at the top. Okay, and it's not difficult to convince yourself that with a spectrum you will have this happening. You must have this happening. All right, so now let's list the last character of every k-mer in the right column. Okay, so there they are. And these were the labels that were on the edges. Okay. These are the labels that are on the edges, and we're going to push them back along the edges to where they came from over here, so that we get a list of outgoing edges for each k-mer, outgoing edge labels. So for this particular k-mer, the outgoing edge labels are A and C. Okay, so we're going to collect these edge labels in that order, the colex order. So these are the sets of edge labels here. And it's this sequence of sets that's the SBWT. So how do we use this sequence of subsets of the alphabet for searching, for doing this k-mer lookup query? So the components we're going to use are the SBWT itself, and then this column of characters, which I've drawn over here, this column of characters, which is just the last character of every k-mer. Okay, and for FM index people, this will be familiar. This column here. This is just the C array, what we normally call the C array. So this says for where the first k-mer having each character, each k-mer ending with a given character occurs. Okay, so C is equal to 8, for example, because at position 8, oh, you can't see my mouse, I keep forgetting that. So C is equal to 8 because at position 8 we have the first k-mer ending with C. Okay, and we have a few of them here, and then at position 11 we have a G. Okay, so this is just telling where those k-mers are. Okay. All right, so we're going to use this small array of size sigma and this sequence of sets to do k-mer lookup. I'll try and give you some intuition for why this works. Okay, the way that search is going to work is that we're going to process our query k-mer from left to right. Okay, so here's a query k-mer, and we'll start by so the goal is to report the colexographic rank of this k-mer. So we're going to process it left to right. We'll start with the first character, and we'll start by finding the colexographic interval of all the k-mers that end with that character. And then we'll proceed by some magic to provide the to find the colexographic interval of all the k-mers that end with AG. Okay, this slightly longer prefix of the pattern. And then we'll extend this again to find the k-mer itself if it appears in the spectrum. Okay. All right. So I'll try and give you some intuition for how this will work over the next few slides. But yeah, so we're processing the pattern left to right, and we're finding this series of intervals. All right, so now first, here's an important property. So if we follow the same edge label from two different positions in the k-mer spectrum, we're going to end up at two different positions. This is by construction. Okay, so there's only one incoming edge to each k-mer. So following the same edge label must take us to different locations. But also let me give you another example. Okay, so here's following two A's, different locations. So we must end up at different locations, but also we must end up at two rows that are in the same relative order. Okay. And this is a direct consequence of these k-mers being in colexographic order. So if they're in colexographic order and we have this k-mer above this k-mer, then when we append the same letter to them, we're going to end up with two strings that have the same relative colex order. Okay. So this shows this in a slightly more abstract way. Okay, we start at two different positions. We follow the same edge label, so we're appending a C to both these places. We end up at different points, and we end up inside this C interval. Okay? So this is the colexographic interval of everything that ends with a C. Okay, now if you consider all the rows that have a C, an outgoing edge label C. This is what the picture looks like. So what we've got, if we've got all the rows that have an outgoing edge label C, they must fill this C interval of colex space. They all go to different positions because of this way we've constructed it. There's only one incoming edge at each position. And they must have the same relative order. So we get the C interval being completely filled. We must get this kind of patterning happening. All right, and now if we consider the rows that share a common suffix, so all these rows share a common suffix, capital A. If we follow the rows with the same edge label, we end up with a subinterval of this C interval. So they must be adjacent, and there must be just as many as we started. Where this subinterval begins is equal to the number of edge labels labeled C above this interval, so in this region. So if we count the number of edges labeled C above this position where we start, we end up with the appropriate offset inside this C interval here. And if we want to know the endpoint, we can also do that by counting. So if we count the number of C's up to and including this position, we end up with the ending point of the interval. So what we can now do is this kind of movement. Okay? We start with an interval that contains everything that shares a suffix. And we append a letter, and we end up with an interval that contains the original suffix plus the new letter. So we're able to affect this k-step search process that I alluded to earlier through these counting queries. All right, and counting edge labels corresponds to counting the number of subsets that contain the letter that we're interested in. So in this particular case, we want to know how many edges are labeled with C in this portion. That's like counting the number of subsets that contain C in the SPWT. So that will tell us the offset inside the C interval. It's two positions inside the interval. And lo and behold, the example works. Okay. So this kind of machinery, I think, is familiar to a lot of us in the room, people that have worked on the FM index for years. But I think it's good to sort of go back, and certainly for young players, to see how this machinery works. I think it's always good. The FM index and Burrows-Wheeler based indexes are one of those things that you kind of spend a lot of time understanding, and then you don't want to think about it again. But I think it's sort of good to go there again every now and then. Okay, so we end up with a search algorithm that looks a lot like the FM index search algorithm the main differences are that we're doing subset rank instead of normal rank, and we're stepping through the pattern in left to right order rather than usual backward order, but this is just a result of the construction. Okay, so the key insight is that we've got these subset rank queries. And the SBWT supports k-mer membership using k of these subset rank queries. And now we have the problem of designing a data structure to solve subset rank queries. So before I talk about one of those data structures, just a word about BOSS. So I asserted before that it's really small, but it's hard to implement. And the main difference between the SBWT and BOSS is that we've arranged for every node to have in degree 1. This was the key thing, that you can take out in degrees and everything still works. And you can do, so you can make it work if you don't do this, but you have to manage the in degrees. And this is the main difference. This was the key insight. And of course, there's a lot more of this kind of thing, as I've been saying. The SBWT you can think of as a specialization of Wheeler graphs for this particular problem, for De Bruijn graphs, actually. And that's the Wheeler graphs are a generalization of the FM index. So we're somewhere in there. All right, I think I'm okay for time. Let's talk about implementing subset rank queries. This is one solution. So this is the fastest solution that we have currently for implementing subset rank queries, and it's simply a binary matrix. It has sigma rows, okay, and it has n columns, where n is the size of the spectrum. And each column just represents the set. Okay. So the set at this position in the SBWT just contains an A, right? So I've got 1 on and we've got the rest of them 0. Here, the outgoing edges are labeled with C and G, okay, and the rest is 0. And we have empty sets as well. Right? This is occasionally annoying when you're implementing things, but it's captured naturally in this representation. Okay, so it supports subset rank in a really obvious way. Right? You pick the appropriate row, and you do binary rank on that row up to the query point. Okay, so binary rank just counts the number of 1 bits in that row, so 2 in this particular case. And we have really fast implementations of binary rank. This is where succinct data structures started. Okay, so implementations in the SDSL, for example, that use a sublinear number of bits on top of the bit vector itself and answer rank queries in constant time and very fast in practice. Okay, so this was our sort of, this is our fastest revved up implementation of subset rank queries. And this is how it performs. So a quick word about this graph. We have space in bits per k-mer down the bottom. And we have query time in microseconds per k-mer, query k-mer. So we want to be somewhere down here. Okay, this is best. This is BOSS as implemented in the Vari tool. So this is the pre-SBWT version of the data structure. And this is where matrix ends up, so it's a lot faster. Just to rub it in, it's more than 100 times faster. So we were really happy with this result. And yeah, so these were using, that's using SDSL bit vectors. There are methods to compress bit vectors as well. So we can use the matrix representation, but instead use compressed bit vector representations on the rows. Like RRR bit vectors, for example, which use entropy number of bits and with lower order terms and still do constant time rank queries. So this, when you apply RRR to the to the matrix, you end up with what you might expect. So you're sort of up and to the left. You get slower and you get a bit smaller. So now we have something that's smaller than BOSS and still quite a bit faster, about 10 times faster. We spent some time analyzing what that H0 compression does to the matrix. And basically, it gets you down to 3.245 bits per k-mer. And the question is this an efficient representation? And that I don't know the answer to, because we haven't been able to count spectra yet. So normally in succinct data structures, you want something that, if you want to know something is an efficient representation, you first try to work out how many different objects you have, right? So if you're talking about a succinct data structure for binary trees, a binary tree on n nodes, so there happen to be about 4 to the n different binary trees on n nodes. So we would need log base 2 of that number to get, at least, in order to represent all the different trees, okay? So 2n bits, and it turns out there are, in fact, binary tree representations that take 2n bits plus a lower order term. This is what's called the information theoretic lower bound for binary trees. So the question is, what's the information theoretic lower bound for k spectra? Or, in other words, how many k spectra of size n are there on an alphabet of size sigma? So we tried to think about this problem. Didn't get really any satisfying place. So what we know is we can represent the spectrum as an n by sigma binary matrix that has exactly n1 set, okay? This was our first attack. But it turns out that not every such matrix corresponds to a spectrum. Here's one that is not a spectrum. So it's n by sigma, it has n1 set, but there's no k spectrum. So this is an open problem, although I'd really like to know the answer to. It would allow us to gauge how good these representations are. All right, so back to experiments. So this is where we left the performance graph, and we came up with some other subset rank data structures, which I'm not going to discuss, which we called split and subset wavelet tree and concat. And these all use quite different approaches, and with trade-offs that aren't particularly satisfying. Okay, this seems like a useful point in that it totally dominates this matrix representation. These ones are starting to get a bit slow, I think, to be actually useful. But what's in this region? And I think there are things in that region. We stopped thinking about different approaches at some point, but actually after we published this paper, but I think there's more to be done there, both engineering and structural design. So it's another open problem. All right, what about compared to these hashing methods that are so widely used? So we compared to 2 different hashing methods. The first is by Giulio, and he'll probably come back at me in question time about this. SS hash, this is the fastest method we know of. The other one, which I think is probably more widely used, is called Bifrost. It uses Bloom filters and other methods that I don't fully understand. So this is the performance graph where we left it. This is where SS hash fits in. So on this data set with these queries, it's slightly faster and about 2.5 bits per k-mer bigger than matrix. This is where Bifrost is, and I just note I had to change the x-axis there, scale, because it's quite a bit bigger. It's about 20 times bigger. And it's about 2 times faster on this data set. And on other data sets, the picture's different. Okay, matrix is a lot faster and it's a lot smaller. So these points do move around, but the general pattern's the same. But I can talk more about this later if people are interested. Okay. I think that's all I wanted to say so far. So for the next few slides, I'll talk about now doing batches of these k-mer lookups. Okay. So as I said earlier, a typical use for a k-mer index is to perform a huge number of these lookups in a single experimental run. Okay, so for example, we might want to look up every k-mer in every read in a metagenomic sample. So that's already talking about hundreds of millions of queries. And they arrive all at once. Okay, here's our SBWT again in black. And remember how lookup works. So we have up to k pairs of rank queries. Okay, so we're asking, by rank I mean subset rank, of course. Okay, so and we sort of do these rank queries in a loop. So we start and we find everything that has T as a suffix, and then we have everything that has AT as a suffix, and then eventually we end up at our. So this is just to bring it back into your mind. And it doesn't take you long to realize that this has poor locality of memory reference. So what I mean by that is that the points at which current rank queries are asked are not close in general to where the next rank queries are asked. So we're getting this kind of jumping around in collect space and jumping around in the associated rank data structures as well. Okay. And this is thought to be an inherent sort of bottleneck with BWT-based structures, not just the SBWT, but FM index, and they all sort of do this movement. All right, and the standard way to process a batch is, thanks Eleanor for this animated slide. The standard way to process a batch is just to run this separately for each k-mer, each query k-mer. This is what we started to call horizontal search. So if we think of the queries as being a matrix, horizontal search is just processing each row at a time, okay? Restarting search each time. And this doesn't improve locality of memory reference at all. Okay, so what we asked ourselves was, can we do better? Is there a way to coalesce these memory accesses that are made across patterns? Okay, and the answer is what we call vertical search or vertical batch processing. Okay. And sorry, all this magic move is thanks to Eleanor. It's great. I haven't learned how to do it myself yet. It takes me off guard occasionally. So with vertical search, what we do is we process a single character from every k-mer in every round. Okay, so search works in k rounds. And in the first round, we process the first character of every pattern. And in the second round, we're going to process the second character of every pattern. And we'll maintain these queues. Okay. So to begin with, we initialize the queues. We have a queue for every alphabet symbol. And we initialize the queues with the patterns that start with that letter. Okay. So here in QA, we have all the patterns that start with an A. And here in QC, we have all the patterns that start with a C and so on. Okay, so this is the way the queues are initialized. And we also store with each pattern, patterns are k-mers, I'm going to call them patterns. We store the collects intervals associated with that particular prefix that we've processed so far. Okay, so everything in the A queue will have this interval, which contains all the k-mers that have A as a suffix. Okay, and everything in the C queue will have all the k-mers that have the area of the collect space that have the k-mers with C as a suffix. Okay. All right, so this is the way the queues are set up. And now in each round, we process the queues in order. So first we're going to process the patterns in the A queue, and then we'll process the patterns in the C queue and so on. And as we process, we're now going to, in the second round, we'll look at the second character of every pattern. Okay. So in processing this pattern, what we do, so the second letter here is a G, second letter of this pattern. So what we do is we issue our subset rank queries on the SPW2. We count how many G's are above here. We count how many G's are above here. This gives us a new interval somewhere down in the G region, okay, of colex space. In fact, it's a region that contains all the k-mers that have AG as a suffix. So we compute that interval and we move this pattern down to the GQ along with the associated new interval. Okay, and we do this for every pattern in the queues in order. So this is the colex interval that we're processing, and it gives us a new colex interval, which we're moving into these queues. All right, this is, what happens here is illustrative, right? So what does this pattern here start with? It starts with AC. Okay. This pattern here starts with CC. We've moved it from the CQ. And so this colex interval that's associated with it, this is everything that's ending with CA. And this will have it associated with everything that's ending with CC. So we've got a colex interval that's here somewhere and a colex interval that comes after it. And what this queuing process that I'm describing enforces is that all these colex intervals will be in non-decreasing order. Okay. So you'll have this beautiful sweep through colex space of the intervals as in the next round as we process them. So I'm not sure how long this animation is going to go on. Probably quite a while. All right. Now these queues have been emptied. These become the input queues for the next round, and these now empty queues will be the output queues. So we've always got these two sets of queues going. We process the third character, and now, as the illustration shows, what we get is these intervals in non-decreasing order. So all our rank queries form this sweep across colex space. It's really nice. We're going to have to wait for this animation, but I think you get the idea. All right, so this is a great illustration of what's happening. This is the memory access pattern, or that sort of sequence of rank queries, rank query pairs, for just 100 queries. And it looks really random. Right? This is for horizontal search. So this is processing each pattern separately. So if I zoom in, you can see there's that large interval at the top, and I get another one. But they're spread all over colex space. And this is what it looks like for vertical search. We were really happy when we saw this plot. Right? Everything's in this nice sequence now, as we'd expect. All right, so does it make a difference in practice? That's the big question. So here's a plot showing query time on the y-axis. This is microseconds spent per query. And this is increasing batch size on the y-axis. So 1 million patterns, 10 million patterns, and so on. And you can see that the time per query for horizontal search is pretty steady. Okay. But for vertical search, first of all, it's less, so even with the small batch sizes, it's faster. But we get this sort of increasing performance as the batches become denser, as the patterns become denser across colex space. So it's about a 4 times improvement. That's on a small data set. And on a big data set, it's even more pronounced. So this is sort of data set of the hour, I guess, this 661K, or maybe, yeah, there's an even bigger bacterial data set out now, but so this is a serious amount of data and queries. And we see an even bigger improvement. This is where SS hash fits in. So It's faster than horizontal search on the small data set, actually also on the bigger data set, but it also has very steady query performance. It's not able to take advantage of the batch size. And you may ask, and it's a perfectly reasonable question, why don't you just sort the patterns to begin with? Maybe this is a sort of cheap way to get some performance benefit. And that's what this plot shows. So here's normal horizontal, here's vertical as we saw, and this is what happens if you first sort the patterns and then run everything. So you get some regularity, but it's not the kind of nice sweeps that we get in vertical search, and it doesn't pay off in practice. You get a little bit of a performance benefit, but not much. All right, so just to say about batch lookup. So yeah, we've improved the locality of memory reference. This comes out. This is a long time coming, right? I think I said at some point this was thought to be inherent, that we jump around lex space or colex space, and we must cop these cache misses. And this is a way to mitigate them. And yeah, so this is nice. I was thinking why I put this point on. Maybe 25 years isn't a very long time. Maybe that's just took that long. Okay, and there is a caveat here, and that is that we have to have the patterns in memory. We're pushing them around in these queues. And we show that you can do that. Like, it doesn't cost you anything in terms of cache and runtime, but you do need that memory there. But then you should be putting the memory to use, right? So I think part of the reason for succinct data structures and compressed data structures is that you can use RAM in different ways. Okay, I think I'm nearly out of time. We've got some other things about batch processing that I didn't talk about. So actually, with a dense enough batch, you shouldn't have the rank structures at all. Okay, you're just scanning the bit vectors basically, and this begs the question: well, what is going on? And it's really that you're merging the patterns with the index, and this might be worth thinking about. We also have something called vertical streaming search, which is allows even faster and search times. And so as future work, we're working on multi-core and thinking about GPU implementations of this stuff. I think batch processing has been sleeping there, and we could use it for other compressed data structures as well. Maybe move, or maybe binary trees, or I don't know. All these things should be sort of maybe rethought in terms of batch processing. Yeah, there's, I think, lots of questions to explore in this space. I thought I'd pontificate for a minute at the end. I think there's a great opportunity for succinct data structures on GPUs. So I think this is where succinct data structures people have been. They're like, well, okay, you can't operate on disk. We can make you small enough to fit in memory. I think that's a real contribution. Then occasionally we say, well, maybe you can fit in cache, but we get these weird cache effects. So that doesn't really work. But maybe I think succinct data structures can get a big enough reduction that we can fit in GPU memory. Then it becomes, okay, how do we query them? We might need to redesign query algorithms to work on GPU. I think there's lots of interesting stuff to do in this space. We implemented matrix on a GPU. Master's student Harry Kakkonen implemented it, and so I changed the x-axis, the y-axis here. So this was where we had the performance graph before. This is GPU matrix. It's a lot faster, and this is like, you know, this is an initial kind of prototype, but I think it says that there's something we should be doing here to try to exploit these computing devices. Yeah, these are apples and oranges. These are all running on a CPU. This is running on GPU, but there might be a reason to try to do that. Yeah, so just a big picture conclusion. I think succinct indexes allow things like batch processing. They might allow us to use GPUs, but they allow us to use RAM in different ways. And I think also we shouldn't become too obsessed with saving space. I think in the early days of succinct data structures, it was all about being small, and people don't find that useful because they want throughput. And so sometimes bigger is better. Well, you always want this trade-off, of course. And almost there. I'd like to thank the Academy of Finland, the University of Helsinki, support also from the University of Queensland, support Joel's visit, and all the great people that I work with in Helsinki, making that a really stimulating place to work. This project and distracting me with other projects. And that's all I wanted to say. Thanks.
On the comments.
Thank you, Simon. That was very fun and clear. I was just going to say back to the information theoretic lower bound, you were saying you don't know how to count this, or we don't know yet how to count the spectra because some of them aren't really spectra, or some of these bit matrices are not really spectra. So a point, which I don't know if is a good point, is that if you just shift the problem statement to be that you're working with k-mer spectra of sets of strings instead of the k-mer spectrum of one string, then I think the matrix is the right thing to count because, you know, one input you could possibly get would be a large collection of k-mers.
Right.
And so I don't know. It sort of takes this fun of the spectrum out of everything, but it's but it does correspond.
It might give you a useful lower bound. Yeah, that's fair comment. Thanks. Yeah.
So thanks for the talk. You mentioned that the similarity with the sorting, the difference. Can you clarify what is the difference between sorting and what you did? Is it like sorting? So you have like the sort of the prefix of each length 1, then length 2, and length 3. Is that
if
correct?
you just take all your query k-mers and sort them, I guess into colex order. It's always does my head. So if you sort them, and now you process the batch in using horizontal search, so one query after the other. So you'll get like a lot of queries for A, but then to begin with, but then they'll start to diverge pretty quickly.
So to clarify, what you are doing is you sort it on the first letter, then the second, and then the first 2 letters, and then the first 3 letters.
Yeah,
right?
but the queries are issued so that they are in order in colex. So it's not. So sorting everything up front doesn't give you the same.
I mean, not everything, but I thought you were. It looked like you were doing like a radix sort.
Yeah, it's like radix sort. Yeah.
And also on the graph you had.
Do you want me to go back?
Yes, please. I don't remember which page, but you had like maybe that. Sorry.
Oh, which? This
next
this
one. The next.
Sorry. Yeah. Okay.
The experiment. Sorry. The experiment.
Yeah.
Okay, it's going through.
You need to be able to shortcut these things in questions, don't you? That one? Next one?
The next. Yeah.
The one with sort?
Sorry. No, sorry. The previous one. Sorry. It's a different question. Sorry. When you for 10 to the 7th, it does on the bacterials. It doesn't change, right? So is this because it's the queries are still sparse?
Yeah, I would say so. Yeah.
So when it gets dense, it means you are searching like on similar ranges, right?
Yeah.
You have a lot of queries on the same range.
Yeah, exactly.
But does this mean that because it's dense, can you not just like go through all the index and
we don't. Yeah, yes, and that's an important improvement. So if you've got the same query as you just did, you should branch and reuse that information. So, like, if you actually reissued the query from a cache point of view, you'd be fine. But there's computation involved, and you want to avoid doing that if you can. Yeah. Does that answer your question? You're asking something else?
I'm thinking. I'm not sure. So.
Okay. We can talk about it later.
Thanks.
Yeah. Cool.
I had another that I think might be the same as Hideo's, which is, could you just add merge sort on this plot? You know, just collects merge sort of the entire
thing. Yeah, I think you would want to optimize that. But I think, and that's kind of what I was hinting at with this comment, is when you've got a really large batch of queries, you don't want an index. Yeah.
Thank you, Simon, for the very nice talk. Now, just one general comment about this, you know, batching query business. Of course, I mean, you mentioned we have to keep the queries, you know, in memory, so they end up in taking, I don't know, a few gigabytes maybe with k equal 31.
Something
and,
like that.
You know, 1 billion.
A lot of memory. Yeah.
So, so I think maybe one can also try to address the following problem. So we're given a memory budget, say, I don't know, like 10 gigabytes, and you can, you know, use all that memory budget to solve, you know, the queries as fast as possible. Because my point is that, okay, so you keep the queries in memory and your index is tiny, but perhaps you can use that space to obtain a, you know, slightly redundant index, which is also 10 times faster.
Absolutely,
right?
yeah.
So,
and I think that's the right setting that we need to think about. We've got a computer system that has a certain amount of resources, and we want to put them to use.
So it could be a nice problem to study. Maybe
I think so. Yeah. That's a very nice way to put it. Yeah.
Thanks.
In all your experiments, you chose K to be 31.
Yeah.
Does a different choice of K make any difference in terms of the space-time plot, or?
Probably, yeah. I don't have that data on hand, but yeah, it's very good, pertinent question. I think we tend to get in the habit of using 31 because a lot of pipelines use that, but then, yeah. So I don't have a satisfactory answer for you, but.
Just a second question. Thanks again for the explanation of the definition of SBWT, because previously I didn't fully understand the definition from the paper.
Okay.
Just wondering, so for BWT, those characters are the left context or extensions of the suffixes. And for SBWT, is there an interpretation in terms of the left of something, or what are those subsets?
They're the outgoing edges from that k-mer in the de Bruijn graph. I think that's the way I think about them anyway. Yeah. Okay. But yeah. I think thinking about it set up like that as this bipartite situation is the clearest way for me to think about it. Yeah.
That's definitely super clear to me.
Yeah. Okay.
Thank you.
Any other question or comments? Okay, thank you, Simon.
All right, so thank you again for having me here. It's a great pleasure. And also, you know, thank you very much, Simon, for introducing all the background that I need. So he mentioned actually many things are directly connected with this talk, namely the Bruijn graphs, why do we want to have fast k-mer lookups, and also he mentioned that sometimes we do not only want to test membership for k-mers, but we do also want to go, you know, a little bit further than that and retrieve some satellite data. And this is exactly what I'll be talking about here. So we'll see what is a color compacted Bruijn graph and how we can compress, you know, this object very effectively. So let me then jump straight to the problem we are dealing with. Again, this will be slightly redundant from the previous talk, but you know, there we go anyway. So we're given a large collection of references that are our favorite references, of course. So under, you know, drawn from the DNA alphabet. And the problem we deal with is that we want to build an index for this collection R so that we can retrieve as efficiently as possible this set here, okay? Which is the set of all the reference identifiers that contains a given k-mer X. And I'm gonna call this the color set of the k-mer. Of course, note that if the k-mer does not appear at all in our collection, the color set must be empty, okay? So this must be done in an exact way. So if the k-mer does not appear, we do not return anything. Of course, there is a lot of hype in the indexing community when this collection R is a pangenome, so namely is made up of, let's say, related genomes, whatever related means. For people doing data structures, related means, highly repetitive, of course. Although, some guys say, well, it's not necessarily true. A pangenome can also be another thing. But, we deal with the case where a pangenome is a set of similar genomes. And this problem is, of course, relevant for, many, many applications. Simon mentioned, something already. So in general, when you want to perform like a pseudo-alignment of, a sequence to, your index, single cell RNA, and so on and so forth. I'm not even, that familiar with all those applications, but I know this is an important problem to solve. And so, our goal is to implement this map from k-mer to its color set as efficiently as possible, in both, aspects. I mean, for both query time and storage and space. And so I'll be using two interrelated data structures. One now we are very familiar with, given my talk from 2 days ago and given the talk by Simon about the spectral Burrows-Wheeler transform. The first one is a k-mer dictionary. Okay, something that stores all the k-mers in our input collection R in an exact way, so that if something does not appear here, we return like an null identifier or something to signal that the k-mer does not appear. So exact way. And the other component is what I will call it, the inverted index or color set matrix, call it as you want, is the collection of all the color sets, okay, for all our k-mers. And I like this view because it's, very elegant, very simple. And as computer scientists, we love this kind of, modular indexing layouts because this allows us to, work separately on two problems. So that, if you improve, something here, you improve, the entire thing, the entire pipeline. Or if you improve something here regarding space and time, you also improve, the entire thing. So I think it's nice, okay, to think in terms of modular, components that you can plug and play and switch. And actually, as a matter of fact, if we take a look of what's there, in the literature, of course, there is a large amount of indexes based on k-mers that were proposed during these years. And actually, all of them are incarnations, let's say, different implementation of this modular layout. Okay, of course, they differ from in the way they represent the color sets. They may use filters, they may use integer compression methods. For this guy, we can use not only SSH but spectral Wheeler transform, the boss. I mean, we can use many different data structures, but this is what has been actually implemented. All right, so yes, so the index I'm talking about here is no exception. So also in this case, we're gonna implement as much in the best possible way these two data structures. And to do so, we have to first understand and exploit the properties of our problem. Because otherwise, if we are not exploiting all the properties, then we're losing, some power. Okay, we're losing some opportunity to boost space and time. So actually, we talked a lot about the properties of this problem already when I gave the talk 2 days ago about the k-mer dictionary data structure. So namely, we deal with k-mers. Okay, so it's a very specific type of string. They're coming from a small alphabet. They have fixed length, and they share, okay, overlaps, okay, k-1 symbol overlaps. So if I take a k-mer set, okay, coming from, by instructing all the k-mers in sequence, there is a lot of redundancy, okay, that we may want to collapse to obtain an efficient solution. And a good combinatorial object that, grasps all those properties is, of course, the Bruijn graph that I'm pretty sure you're already familiar with. So this is a slight repetition from 2 days ago. So this is the Bruijn graph for k = 3. So as you can see, we have one node for every k-mer in our dictionary, and we have a direct edge between two nodes if the suffix of length 2 of this guy is equal to the prefix of length 2 of the other guy. So it's a very actually there is a one-to-one correspondence between the Bruijn graph and a set of k-mers. Okay, so I can talk about one thing or the other one. It's the same, and there is a lot of redundancy, of course. Okay, so namely we can collapse the k-mers along the non-branching paths. But before going there, so let me first explain what the colored keyword means in this context. Well, of course, it dates back to, I guess, the first paper who introduced, that introduced the concept of colored Bruijn graph. So they were referring to each reference in our input collection as having a color. Well, actually, as being a color, like red, the color red, blue, yellow, and so on. And so, using this formalism of the Bruijn graph, if I build the Bruijn graph out of the entire set of k-mers in my references, I can see each input reference as a path in this graph. Okay, so those paths corresponds to the original references. So I have a one colored path for each of my references. But that's not really important, okay? So that's like the pictorial, like intuitive, let's say, visualization, okay, of the object. So now let's see how we can index this object. So first step, we get rid of this, colored lines. We don't really care. We logically, to represent those lines, we logically annotate each k-mer with its color set, okay? For example, I don't know, TTG is annotated with 3, 4, 5, and 6, because of course, as you can see, it's crossed by reference 3, then 4, then 5, and 6. Okay, so we do this. Okay, we do this logical annotation for each k-mer, and again, you can immediately notice. Well, this is a toy example, okay, but if you think in a bigger scale, you can immediately see that there is a lot of redundancy. And namely, k-mers along the non-branching paths happen, most of the time to have exactly the same color set. If here this is not the case, we break here. We break the unitig here. And so we, as you can see, we merge k-mers along the non-branching paths that have exactly the same color set. Okay, so as highlighted by those red boxes. So for example, these three guys can be glued together in a unitig because all of them have exactly the same color set, the same here, same there, and so on. So we're now reduced to this. And so now we have an equivalent colored the Bruijn graph, which I will be calling the compacted version, of the graph. So now each node, it's a unitig. Okay, so those longer strings are called unitigs, and we have this special property by construction. Those unitigs are monochromatic. So we say that these are monochromatic, meaning that if I look at each k-mer inside a unitig, they have exactly the same color set. Okay, so unitigs are monochromatic. So this is the object now we're gonna index. So again, talking a little bit more about the properties of this colored compacted Bruijn graph. Some of them have been already mentioned several times, so I'll be quick on this. But first property is that unitics spell references in R. So it's a lot more convenient, of course, to store the set of unitics and not the set of the original k-mers for saving space. So here is the set of unitics from the previous example. So I would like to store this set. And again, what is a nice k-mer dictionary that can store any spectrum preserving string sets like the unitics? SSH. So I'll be using SSH here. Right, so with this, we again spend space proportional to the number of unitics and not the number of k-mers. Property 2 that we just mentioned, unitics are monochromatic. So I can logically associate again one color set for each unitig and not for each k-mer. And of course, there are way fewer unitics than k-mers. Okay, so that's already a big reduction in space. And what I need now is this map. Okay, so I need from a k-mer X to go to the unitig where it appears in. And there are, again, several solutions to implement this map. But since here we are using SSH, for SSH is very easy, okay, to do this map. So let me quickly say again what this is. So given a k-mer, I want to identify the unitig that comprises that k-mer. For example, I don't know, if I take TCC, okay, I look up for TCC in SSH, and SSH will end up in locating this unitig number 4. Okay, so unitig, so the result of this unitig query for TCC as well as for CGT, I mean for every k-mer inside here will be identifier 4. So now with this number 4, I can directly look up in this color set matrix in this inverted index L and directly retrieve the color set number 4. Okay. But of course we can do better because again you may notice that. Can you see the colors? Yeah, more or less. Okay, I mean, you can see that these two color sets are the same. Also, these two are the same. Again, this is a small scale, really a toy example. But again, in reality, okay, we have many color sets that are actually the same. Okay, so we would like to index the distinct color sets. This happens because unitigs co-occur, very frequently. And this is actually very intuitive. Why? Okay, because the original collections being a pangenome, so formed by related genomes, those genomes happen to be very similar. Okay, so this has been mentioned already many times during this workshop. If we give a look at my genome and yours, it's going to be almost the same, apart from a few, a few bases, or I don't know. So we're gonna share, okay, a lot of unitigs. Okay, that's the point. And so so we want to store the set of distinct color sets. And so now my problem it's a little bit more complicated because I need to implement this map, okay, from the unitig ID to the distinct color set ID. And here is where I exploit this nice property of SSH that I can rearrange the order of these strings. So I can permute the unitigs as stored in SSH to boost some compression for the satellite data. And guess how I'm gonna permute this unitigs. I'm going to permute this unitigs so that consecutive unitigs have exactly the same color set. Okay. So for example, so we saw before that this guy, now it's a bit complicated to see. I don't remember anymore. So where do was? Okay, So this guy and this guy had exactly the same color set. And so now I want to put them, in consecutive order. It's, quite easy, okay? But, we have this. We don't have many distinct color sets. We just 8, and before we had 10. But, across large collections, we observed that we have like 2 times less, maybe 3 times less sometimes distinct color sets than unitics, okay? So it's important to represent the set of distinct the set of distinct color sets only. All right, so now I'll be representing this, okay? This permuted set of unitics and this color matrix made up of distinct color sets. Of course, now I have this problem that I need to map, unitics to the same color set, but also this can be done in a very simple and efficient way using rank queries that also Simon mentioned, previously. Okay, and so we have very, efficient solutions for Succinct rank. Okay, so I can just build this bit vector. Well, I have 1 bit for each of my unitics, and you can view this in many ways. You can say that you write the number of unitics that have exactly the same color sets in unary, or you can say that you mark with a 1 when you have to change the color set. You can view this in many ways. The point is that from the unitic ID given by SSH, I can do a rank, okay, from this position, and this gives me exactly the color set ID. So this can be done in a very efficient way. So rank, as Simon said, it's like 30, 40 nanoseconds. So it's very, very fast. So we implemented all. One final, word. Of course, to compress now the color sets, which are sorted integer sequences, you can use essentially, your favorite compression method. So you can refer, for example, to this survey where I and Rossano, surveyed, many of the techniques that can be used to compress inverted indexes. So any method here would work. It depends, of course, on your target application. The wanted level of performance you want to achieve. So namely the trade-off you're interested in. For this first work, we are gonna opt. I opted for something very simple but effective. So I'll be using 3 different encodings based on the density of the color sets. So if C is the color set and N is the total number of integers, so the total number of references that I have. So if this ratio, goes below 1/4, I consider the set sparse because it contains, a few integers. And so I'm gonna use this simple trick, very folklore. I take the gaps and compress them with an instantaneous code like Elias delta. If instead it's, this ratio, this density is, large, so greater than 3/4, then the set is very dense. And this case actually happens very, very often. So if you index genomes, this case is going to happen, very frequently. And so it's more convenient now to reason in terms of the complementary set, which by definition now is sparse. Okay, so I take the complementary set and I code like in the very same way that I said before. Otherwise, if we do not fall into these categories, then the set is dense and I can just use n bits. So it's a good match. And actually, you can also work out a little bit of math and show that these 2 ratios are optimal for given these 3 encodings. So we implemented all these ideas. So the fact that you can permute unitics, the fact that unitics are monochromatic, Succinct rank, and these compression schemes into a tool that we called Fulgur. Fulgur is a color compacted Bruijn graph index. So namely, a couple of years ago, 2023, Then we had a journal version of that work in 2024, beginning of 2024. And so this is more or less, the results. So Fulgur is here. So it's faster and smaller than our direct competitor, Themisto, that was also mentioned by Simon before. Themisto is also another color compacted Bruijn graph index. It's also a lot smaller than Cobb's, something that was published at Spire, I think. They got the Best Paper Award. And, back at that time, it was a really nice index, combining Bloom filters, SIMD, hashing. So it was really, really nice. It was actually the paper that got me interested in computational biology. So I owe them a lot. But they are huge. Okay, they're huge and also very, very slow. And Metagraph is also, something that we had to compare against. Initially we didn't compare against Metagraph because Themisto, in the Themisto paper, you compared against Metagraph, and they found out that Themisto was already faster and smaller and so on. So I said, okay, then we can skip the comparison. But then Metagraph people got angry, and they wrote me like, no, you should compare against us. I see no reason. Well, say, okay, then let me do a comparison, and I couldn't run the code, and so I asked them, if you want me to run your code, you have to tell me how I can do this. So they sent me like a huge script, and so this is reproduced using their own script. So I'm not cheating in any way. And so, the thing is, their space on disk is small, okay? So it's small, so it kind of looks like they're winning, but of course the query time is prohibitive. Or you can use Metagraph in this batched way, where the query time, it's definitely better. Okay, it's not like hours, but it's better. Still, both Themisto and Fulgur are way faster, but their RAM at query time explodes. They decompress portions of their index, and they expand like manifolds. So I with all respect, I think their trade-off is not as good as what we have in Fulgur and Themisto. All right, so this was version 1. Hey, hi, Dominic. But yet there is another cool property that we haven't exploited yet. And actually, when we were publishing Fulgur, I knew already in my heart that we should go higher order and exploit this property. That color sets are, of course, quite similar when we index genomes. And so instead of compressing each color set individually, well, of course, we can achieve much, much better compression if we go higher order and we, identify in some way common patterns that are repetitive across, the entire collection of color sets. And of course, we compress those patterns only once. Okay. So next, we're going to see, some examples of this. So how can we, how we can capture those patterns. Can you see the colors again? I don't. Yeah, it's very. It's complicated, but on my screen they're very bright and Anyway, so you can see that, for example, we here have 3, 5, 9, 11. Maybe also and these patterns also occurs in this color set and also in this color set, or this other pattern 1, 3, 11, 12, 13, 14, 16 also appears twice. So now my point is, if you encode color sets individually, you're going to repeat the encoding, so you're going to waste of course space because you represent twice or 3 times, as many times as the pattern appears. You're going to repeat its representation. Or these two guys, the 3 and the 4th, are exactly the same. The C3 has a 10, which is not here. This guy has 13, which is not there, but these are more or less the same. So we want to exploit this. There could be, okay, so how do we factor out this redundancy? Well, there could be many possible approaches. The one that we took is to use partitioning. Okay, so we partition the set of colors, the set of color sets. Initially, we, I proposed, to partition them vertically, but then I thought, well, if vertical works well, then maybe we can also, do horizontal partitions, which is also conceptually, easier to grasp. So horizontal partitioning, as you can see, it's like, we group, okay, color sets that are similar, okay, in some way, and we try to compress all of them together in each group. Vertical partition instead, try to permute the columns, okay, in a way that consecutive columns are, of course, similar. Okay, that's those are the two intuitive ideas. So let's see briefly, the representation that uses those two partitioning approaches. So this is for horizontal partitioning. So again, you can imagine that we group, okay, in some way the color sets that are similar. So logically, all these 4 unitics map to these 3 color sets that are similar, and these other here. So we have 3 groups, okay, in this example. And the idea is we look at those color sets within each group, and we're going to synthesize. So we're going to create a color set that I'm calling the representative color set that somehow, really represents, okay, what's there. Okay, that represents, the repetitiveness that is present in the color set in each group. Okay, so namely, you can imagine that, the integers there are drawn, okay, from each color set. So these guys are the most repetitive integers, okay? So they capture, the repetitiveness that is there. And now that I have synthesized this representative color set for each group, then I can express each original color set as the symmetric set difference with respect to the representative set, okay? So I can take the set difference, and the idea is that if these representative color sets capture well the repetitiveness that we have in the group, then those differences are small. Yeah, Simon?
Sorry, so these representative color sets don't actually have to exist as color sets.
Exactly. Yes. Exactly. Yes, good point. So these are not necessarily color sets. Well, actually, I don't think they. Yeah, it never happened. Well, it could be, but sure. But yeah, exactly. So they are never color sets. So these are like a meta, some metadata that we add. Yes. So they work like dictionaries. Yes. Yeah, so the intuitive idea is that these differences, okay, are going to be small. So you end up encoding fewer and fewer integers in your final index. The other approach instead, it's vertical partitioning. So this is slightly, more involved. So let me go through an example. So now we're, partitioning columns, okay, not rows. So we're going to start with, of course, grouping the colors, grouping the columns. And imagine that we identify these 4 groups of the columns. So, for some reason in this example, I group together column 1, 12, 13, 14, 16 in one group. Then I group 3, 5, and 9 in the second group, and so on and so forth. Okay, I mean, those are random. Well, yeah, maybe not random, but more or less random. Just an example. So if I reassign identifiers to the columns following this order, I obtain, a permutation. Okay, which gives you that, I don't know, what was before column number 3 now is number 6. What was before color number 4 now is 12. Okay, so it's a permutation. And so here I'm coming from my background in IR. So this is known as the document ID reassignment problem. So I'm reassigning document identifiers, but it's not important. What's the name? Anyway, I have this permutation. So if I apply this permutation to my original collection of color sets, I obtain this new collection. Okay, where the vertical bars here represent, the partition boundaries. Okay, so namely 3 is here. Then we have a bar because 3 belongs to the first group. Then we have 6, 7, 8 that belongs to the second group. Okay, so those vertical bars are just for visualizing the different partitions. And now what I can do is to look. Yeah, okay, so let's consider partition 2. Now I can look within the color sets of partition 2. So the color sets only relative to partition 2. And as you can see, again, or more or less you can see, that we have 3 distinct color sets. Namely, we have this yellow color set, we have the green, and we have the blue. Okay, so only 3 color sets that I'll be calling them partial color sets. Partial because they are relative to a partition. Okay, so we have only these 3 distinct color sets. And again, the intuition is that if we again look at the distinct color set within a partition only, then these are very, very few. Then these are very, very few. Much fewer than, of course, the total number of distinct color sets. Then, of course, there is a little trick to actually spend less space. Namely, I know that all these guys are coming from partition 2, which starts from identifier 6. So, of course, I can subtract the previous partition lower bound. There are a bunch of tricks to squeeze space even more, but this is the general idea. So if I again do this game for all the partitions, this is the overall picture. Okay, now the original color sets are now represented into using 2 components. So I have the collection of partial color sets. So in this case, I have 4 partitions. Okay, so 4 collections of color sets, and those are like like dictionaries. Okay, that we use. Okay, those are the patterns that we use to reconstruct the original color sets. And the other thing is what I call the meta color sets. They are nothing but pointers. Okay, those are references to those partial color sets that give you like the spelling of the original color sets. I hope this is clear, but just to go through a simple example, if I look at this first pair 1, 4, this means that the first pattern I have to consider is pattern number 4. Okay, number 4 from partition 1. Okay, and the same for all the others. Okay, if I look at 3, 2, I have to jump to partition 3. Here it is, and look at pattern number 2. Okay, so with this thing, I can spell all the original card sets using a restricted number of patterns that are highly repetitive. Okay, so this was the other example, and so with this, we released another version of Fulgur. So we had another paper about this compression-aware, repetition-aware compression of color sets, and without going through all these numbers, just consider this row here, which is the largest collection in this table. So this is for 150,000 Salmonella genomes. So in blue here we have the original Fulgur index, so what we saw a couple of minutes ago. So this was 71 gigabytes, including also the space for the dictionary SSH that takes 2 gigabytes. The card sets themselves, it's 69, but let's say this is 71. So So defulgur is the differential thing, so the thing that computes the symmetric set differences, and we get down to more or less 18. So, fair reduction in space. But if we consider the meta-partial color, so the thing that I just explained, then we go down by 1 order of magnitude. So it's really, capturing and factoring out all the redundancy. Yeah. Those are gigabytes. Oh, sorry. Yeah. Yeah. It's not reported. Yeah. No, because I erased the caption. But yeah. Yeah, those are total gigabytes. Yes, absolutely. Yeah, so it's the final size on the disk. Yeah. And actually, another thing that I didn't mention, but it's so kind of straightforward now that we have two schemes. So the defulgur and enfulgur can be combined. Namely, you can compress the collection of partial colors. Actually, you can even go recursive, maybe, but that doesn't make a lot of sense to me. Or you can use to represent those the differential approach. So you can actually combine the two, and if you do that, you squeeze even more. Okay. Actually, again, this is the total size on the disk, so it's this number. So what we spend for the color sets plus SSH, okay, plus this redundancy here that of course doesn't change, okay, so it's like a fixed constant that it's always sum to the final space. So actually, we should actually compare this guy with this guy, okay, so color sets with color sets only. So yeah, so from 5.3 we get down to 3.2. Again, combining those, we reduce even more. Yeah, so you can see, from 71 gigabyte, we go down to 5 gigabytes. So, and yeah, and another way of visualizing this, well, actually, this is, I decided to put this slide because people, of course, outside, this field, typically ask me, okay, yeah, but why were you still interested these days, in working on compressed data structures? I mean, isn't that a solved problem now that we have neural networks and we can compute, smaller codes for everything? Isn't that done? I say, well, I mean, look at this this picture, for example. Okay, so looking at this slide, this is a nice way of motivating our research, right? So with the fulgur index, you can spend this amount of money, okay, because it doesn't fit, okay, here or there. But if you compress more, of course, now you can fit, within smaller memories, and so you pay Amazon, of course, less, okay. So yeah, so this is a nice way of explaining our research. Of course, not to you guys, but to people working in other fields. Yeah. So we really save money. And okay, so let me conclude. I have 5 minutes more, right? Yeah, maybe. Okay, so just final words to conclude. Of course, as it usually happens working, on compressed data structures, when you compress a lot, you also typically introduce some overhead, some slowdown in the queries that you're performing. And so again, fulgur is no exception. Okay, of course we compress a lot more, but we also go a little bit slower. Actually, not that slower in this case. Okay, so 53 minutes against 44. Oh, these are times in minutes, and these are times for doing pseudo alignment, also the process Simon was referring about before. That is, there are many types of pseudo alignment queries. Here I'm doing essentially at its core intersection of color sets. Okay, so we retrieve all the color sets of the k-mers from our query and we intersect them. Right, so apart from the M fulgur, which stays competitive, then the D fulgur and MD, of course, suffers from slowdown. But we have an upcoming paper at WABI this year that my student is going to present in Washington, I guess. That actually, let's say, introduces a bunch of useful tricks, of course, formalized into proper algorithms that show how to exploit, those patterns to even accelerate query time. And this is now the bigger picture, Now we improved a lot. Actually, this got a massive speed up, as you can see, from nearly 2 hours to half an hour. And so, yeah, so this is also another way of visualizing the improvement. The blue bars are, the original query times. Now we have the yellow bars, current query times. So we improved a lot. So this is a nice way of, showing the power of, repetition-aware indexes. Not only for the space, but also for the query time. Yeah, there's a quick slide about the intuition, but maybe I'll skip. So, again, we'll have a paper published soon. I mean, it's already available on my webpage, but now I'd like instead to conclude showing you this plot that, well, these two plots. The first one is a space improvement plot. The other one is for query time improvement. So the space improvement shows that the more the collection becomes repetitive, the smaller the index, is. So these numbers are the ratio between the space of fulgur, which is not repetition aware, and the space of the full, like MD fulgur, which is fully repetition aware. So this means that if you, I don't know, index 150,000 salmonella genomes, you have a final index which is 15 times smaller. But if you keep adding things, for example, for 300,000, you have 22 times smaller index. And the nice thing that you see is that this keeps increasing. So this is, I think it's a very nice way of showing the power of repetition-aware compression and query time. For the query time, on this large collection, we actually go down a little bit. I mean, I don't know why precisely. Maybe it's an artifact in the queries. So why now we have a slide going down, but at least we keep, a factor of 2 better query time. So, so I'm done. So these are the takeaway messages. So back in 2022, I realized that we can use SSH to map very efficiently k-mers to Unitigs. We have also other alternatives, but that this was back in 2022. Then the year after, then we exploit this property of SSH that we can permute the Unitigs to efficiently map Unitigs to car sets. So this was 2023. Then in 2024, we say, well, actually, we can even factor out the patterns in the car sets. So we introduced this repetition-aware compression of car sets. And now in 2025, we use these patterns to go, much faster than before. So with this, I conclude. Of course, I want to, say thanks to all of you for listening, for the patience, and a special thank also to my co-authors, both Rob Patro and Alessio, who is the student who led some of the latest development in the fulgur index, and is going to also to present the paper at WABI in late August. About the next goals, well, we would like to index more and more, larger and larger collections. The group in Cambridge, led by Zemini Iqbal, are releasing, a new version of these 661K collection called All the Bacteria, where you can see you have many, genomes that we would like to index. So this is probably what we're going to do in the future. Up to the point, in my opinion, we're going to need, some shard into external memory. That seems like an inevitable feature because, all the things that we have been discussing, me and Simon, are for in-memory processing, right? So we want to have an efficient index to do all the, lookups in internal memory. But of course, doesn't feel like something we can, really keep up. I mean, even with repetition-aware compression, if you want to like scale to all the NCBI or all the bacteria, or I don't know, the whole RSA, then chances are, it's very likely that we're gonna split at some point the index into shards, work in external memory, like load those shards and combine. I don't know, but this feels like it seems like inevitable in the future. All right, that's all from me. Thank you. Thanks.
Any other questions or comments?
Thanks, Giulio, for the talk. I had a couple of questions about representing sets. So I've run some experiments using this recursive graph bisection to reorder color IDs, basically, and seem to give me some compression. I like quite a nice boost. Yes. I can't remember exactly now, but I'm just wondering whether, how that fits in, whether you sort of see a place right here.
Yeah, exactly. Very good question, very precise. Thanks. Yes, so I did mention this, quickly, that, so here we have this permutation, okay, which is also known in the IR community as, reassignment the documented IDs. So for inverted indexes, this is highly effective. So part of the, reduction in space also comes from reassigning the doc IDs, but not that much. Actually, yeah. So one thing that I haven't tried yet, but my, suspect it's not so effective is that we can run, for example, some reordering inside each permutation. For example, the BP or reordering algorithm. There are many heuristics. Actually, the problem is NP hard. So there is no way of doing this like, optimally. But there are some excellent heuristics. Yeah, to do that.
So so this is maybe a related question then. It seems like there's a lot going on, but maybe it's simple to implement. But what's the construction time for doing all this? Is it a serious overhead?
Well, or let's say in the first implementation, it was a lot slower and a lot. Now, actually, we have a much faster construction algorithm. It's not a big deal to build. Because for doing the partitioning, we are using sketches and then clustering, which can be done in a highly parallel manner. So it's not a big deal. One thing is that first, to obtain this meta partial color representation, for example, first you need to build a pre-existing Fulgur index. So first you have to build Fulgur and then partition that. And maybe one can also avoid to first build that and directly try to output this. Yeah, but the thing is, I don't have the construction times right now, but for partitioning, this the index over a large collection takes like an order of minutes. Okay. So it's not a really it's not a big deal. Yeah.
I had one more question.
Sure,
which was a paper by, I think it's you and Alistair Moffat, maybe Matthias Petri as well, about dictionary compression in a web search context.
Sure.
And I was wondering, I've never read that paper, but I've always meant to.
No problem.
I was wondering whether there's an overlap of ideas here, and And,
Yeah.
and if not, what makes, so this is like an inverted index for genomics, right?
It is.
What
It is.
makes web search inverted indexes different to genomics inverted indexes?
Yeah, that's also a good point. Right. I think they have like different shapes. I mean, if this would have been for like natural language data, the dictionary would have been a lot smaller. But yeah, exactly. So not so many terms, along this line, but way longer, inverted lists. So, but in fact, I, suspect, I, came somehow from that area that I'm very happy to have actually abandoned. But for other reasons, they're not relevant here. But no, but really, a lot of methods developed there can also be adapted and used here. In fact, my thing, and I was also chatting a little bit with Joel, about this. One should bring these two communities together, right? Because it feels like a lot of problems that actually we do here and they do there, they can also be used together. Also the, yeah, exactly. Yeah. So yeah.
Thank you for the talk. And now I'm really looking forward to the Wabi talk too.
Yeah, of course. Yeah.
When you were designing these schemes, were you operating under the assumption that there are some colors that are going to co-occur in color sets more often than others? Like, is that kind of your working hypothesis for why some of these compression schemes work? Or do you think they work even if co-occurrences are completely random?
Well, I mean, so we experimented that if the collection we are going to index is very diverse, for example, you mix I don't know, many different bacteria genomes, then of course the compression is not that great because the genomes themselves are different. So of course you have to index like a highly repetitive collection. So I don't know if that answers your question.
Sort of. I mean, because a collection can be highly repetitive, but not have any particular expectation.
No, well, I
that
mean,
colors will co-occur.
we don't work under any particular hypothesis. I mean, I mean, if that is your question, I mean, I don't assume.
Yeah.
No, I don't assume anything. But it's like an effect that if we give a look, maybe there is also here the space. I mean, yeah. So if we give a look at these numbers, no, I mean at the progression in these numbers, where let me. No, okay. From 50 we go to 100, and more or less we double. Okay, but from 100, if we go to 100,000, sorry, 150,000. So we do not double, but still the space grows like super linearly, no?
Yeah.
So this is why we have more and more combinations of color sets, and so we spend more space. So. So this is something that I observed, no? That the scalability, in space is not like linear in the number of references that we have. So this was my, that thing captured my attention, and then I thought about, partitioning them. But no, I don't work under any particular assumption.
Thank you for the talk. I'm not sure what I want to ask, but I'll try to make a question.
Whatever.
But, so you said you didn't make any assumption. But basically the problem you want to solve is compressing a family of a family of set of integers.
Exactly. Yeah.
And so if there's no assumption, then any compression algorithm for this would work, I guess.
Yeah, in fact, as I mentioned before, you can pick your own favorite algorithm from here, and you can get a nice solution. But still, you're not exploiting the fact that you have patterns that co-appear, that co-core.
So what I'm trying to understand what kind of the path, what are characteristics to the data? I guess for this kind of data you're using, there's some kind of
the fact
repetition
it's simply the fact that the genomes that we are indexing are very, very similar. So if they are similar, it means that, for example, a unitig appears like almost everywhere. And if it appears almost everywhere, then you're going to have a color set that actually contains all the identifiers. Does it make sense? Okay. So if this is something, I hope quite intuitive, no? If the original documents are very similar, then this similarity induces, clustering in the final integer sets that you're going to compress.
So this means you're going to have the same integer in a lot of these sets.
Yes. Yeah. Yes. And in particular, we have a lot of patterns that are shared across color sets. Yeah, exactly. So subsets of those integer sets that are highly repetitive.
So when you want to choose a good way to compress these, you also mentioned that you wanted to do some operations on them, like set intersection. So there, I don't know about so much, maybe Yasuo knows. So so there's like decision diagrams you can use to represent set of sets, and then you can take intersection and stuff. So.
Decision diagrams.
But yeah, I was wondering. Yeah, sorry, it's not maybe a question.
No, but it's
I
fine.
wanted to understand.
Probably there are also other representations that could be used. Absolutely. We also tried, for example, repair. Which feels like maybe a good match, but it didn't work that well. Also, its construction time, it's terribly slow. But also we thought it's a very good algorithm, and to capture like the repetitiveness that we have maybe can be used. Maybe it's an artifact of the implementation, but it's not as good as what you obtain. As what we obtained. Yeah. But yeah, probably could be also other schemes.
Thank you very much.
Thank you.
Thank you for the talk. Could you go back to the previous slide with the threshold of quarter and 3/4?
Yeah. Okay. With the, I think you.
Here, yes.
This one.
Yeah.
Okay.
Thank you.
Yep.
Just double checking. Here, the definition of sparse and dense is based on the number of distinct colors. It's based on.
No, it's based on the size of the color set.
Oh, sorry. Yes. Okay.
Yeah. So C is a set. So this is its size or cardinality, and this capital N is the total number of integers that you may have. It's total number of references. So the size divided D gives you like a density. So it's a measure of how dense or how sparse the color set is. So if it's sparse, it means that you only have a very few integers there, no? Like I don't know, you have 3, then you may have 100. It's super sparse, and so so you choose like a different encoder depending on the sparsity. This is also like known tricks. It's not like the end of the world. Yeah.
Okay. And so the optimality is proved somewhere.
Yeah. So you can use some bounds using, well, there are actually only for the Elias delta to show that given these 3 encodings, you can choose this threshold to to be optimal. I mean, by optimal, I mean that if something is, for example, here, now it's sparse, if you use this encoding, you lose bits. Okay, that's the idea. Yeah.
Thank you.
Okay, thank you.
Thanks.
So I'm Tomohiro from Kyushu Institute of Technology. I'm going to talk about SLP encoding. This is a joint work with Akito Takasaka, who had already finished his master course and left the university. And this talk includes what we presented in Spire 2024 and what we are adding to the further journal version. So first of all, a straight line program, SLP, for a string T is a context-free grammar that derives T only, so which can be seen as a compressed representation of T. For example, if you have an SLP having these production rules with starting variable x1, we get this string T by expanding x1 according to these production rules. So basically, these rules say that x1 is replaced with x2b. So x1 is branches to x2 and b, left and right. And x2 is replaced by a and x3, and so on. And this is the parse tree of the SLP, showing its expansion process visually. So in this talk, we use this notation. Sigma is the alphabet size, the number of terminal symbols. And large N is the length of the string T. And small n is the number of variables of the SLP. And h is the height of the parse tree. And the string derived from variable x is denoted by this. We also assume that the SLPs are in a normal form, that the right-hand side of any rule is of length 2. So these production rules satisfy this assumption. SLPs are a popular framework to model the outcomes of dictionary-based compressors. Also, there are extensive studies of algorithms to work directly on SLPs, such as self-indexes, pattern matching, and so on. So in this talk, we consider the random access problem on SLPs, which is one of the most fundamental tasks. The task is to compute TP for a given position P on T. So P is the position on T, and TP is the pth character of T, without expanding the whole text T. For example, given position 9 in this case, we compute the 9th symbol B of T, which can be done by accessing to the 9th leaf of the parse tree. So from left to right, these are the positions. And the ninth position, ninth leaf of this pass tree is B, labeled by B. So this is the ninth character of this expanded string. But we want to do this without expanding the whole tree. There is a simple algorithm to support random access in time linear to the height of the pass tree. If we have constant time access to the length of the derived string of a variable, we can navigate to the target leaf on the pass tree from the root. That is, for each node on the path, we can compute the child that contains the target leaf and the relative position in it, like we do binary search on binary search. So here, H can take a value from log large n to small n. Large n is the string length, and small n is the number of variables. And H, the height of the pass tree, is in between these values. Of course, the height H depends on the shape of the pass tree of the given SLP. But if H is close to small n, the random access takes of small n time in the worst case. So question is that, can we improve this time for any SLP? There are two existing approaches to achieve all log large n time random access, regardless of the height of the pass tree. Firstly, Bruijn et al. Presented the data structure for the task, which is built in addition to the SLP. On the other hand, Ganardi et al. Showed that any SLP can be converted to a balanced grammar of height of log large n without increasing the size order. So after converting the SLP to this kind of balanced grammar, the simple algorithm is enough to achieve of log large n time random access. So both approaches work in of small n Warsaw space, but its hidden constant have not been analyzed, and it is probably not small. So in this work, we propose a new SLP encoding for this of log large n time random access using this amount of bits. There are some other related works. Firstly, Tabei-san and others showed the lower bound of this amount of bits to store an SLP. Note that if we want to store an SLP only without random access, it is enough to store the information of the right-hand size of the production rules. But storing them naively takes twice as large as this lower bound, which makes a big difference in practice. And Maruyama et al. Presented an SLP encoding for of H time random access of this amount of bits. And there are some other practical SLP encoding with of H time random access. And Varbin and Yu studied lower bounds of random access time, and showed that any data structure of poly small n space needs this time for random access for any small constant epsilon, which means that O of log large n is almost optimal in general. Now I introduce symmetric centroid decomposition, which is used in our SLP encoding. Firstly, the path tree of an SLP can be represented by a DAG, where there is a one-to-one correspondence between internal nodes and variables. For example, this path tree, we have two occurrences of x5, variable x5, and they are merged into one internal node in the DAG. So in the DAG, each variable has, there is exactly one appearance of variable as an internal node. Then symmetric centroid decomposition is a path decomposition technique for DAGs. For a node U, we consider two variables pi u and pi prime u, which are respectively the number of paths from the root to u and u2 leaves. And lambda u is the pair of log pi u and log pi prime u, which is written in the node in this figure. And for example, lambda of this node is 1, 1, because the number of paths from the root to this node is 2, this path and this path. And the number of paths from this node to the leaves is 3, 1, 2, 3. And taking logarithm of base 2, we have lambda value 1, 1. So these are the lambda values. For each node, it has its lambda value, pair of integers. Now, an edge UV is called an SC edge if these lambda values of U and V match. So lambda value of U and lambda value of V is the same. The edge is called SC edge. And otherwise, it is called non-SC edge. So in this figure, SC edges are shown by solid arrows, and non-SC edges by broken arrows. So these are the SC edges. This one is also, and others are non-SC edges. By definition, every node has at most one outgoing SC edge and at most one incoming SC edge. Then a maximal subgraph connected by SC edges becomes a path, which we call an SC path. Now let n prime, small n prime, be the number of non-leaf SC paths in the DAG. In this figure, these three SC paths are counted in n prime. So red one and the blue one and green one, green ones. Now it is clear that this n prime is at most small n. A notable property of symmetric centroid decomposition is that every root to leaf path traverses at most 2 times Lagrangian non-SC edges, or equivalently +1 SC path. This is because the condition for a non-SC edge requires that at least one of these inequalities hold. So this is an abstracted image. We choose some leaf, and there is a path from root to this leaf. And this path contains some SC path. This is SC path, and this is another SC path. And going through this path, we have at most 2 Lagrangian non-SC edges, or +1 SC path. Because along this path, log pi values move from 0 to Lagrangian, and log pi prime values move from Lagrangian to 0. So there are at most 2 Lagrangian chances to happen this inequality. So using this property, now I will give an idea to achieve all Lagrangian time random access. Recall that our goal is that given position P on T, to find the Pth leaf from the root on the DAG in all Lagrangian time. Now suppose that there are E non-SC edges along the path from the root to the target leaf TP. Starting from the root x in 1, we leave this the first SC path at x out 1, and land at x in 2 in the second SC path, and leave the second SC path at x2 out, and land at x3 in the third SC path, and so on. Now we set our subgoal as to move from x in I to x in I plus 1, namely one SC path to the next in this time, depending on the difference of logarithm of the derived string length. If this subgoal is achieved, the total time for random access is the sum of this value, which results in of Lagrangian, because these plus log terms are cancelled by these minus log terms. This is kind of a telescoping argument. So we get this of Lagrangian time. Is it clear? So now we focus on a single SC path with M nodes. So in this figure, we have an SC path of length 6, having 6 nodes. There observe that there are M plus 1 non-SC edges branching out from the SC path. So let V1 to Vm plus 1 be the endpoints of these non-SC edges. sorted by the pre-order of left to right traversal from U1, so V1 to V7 in this order. The string derived from U1 can be obtained by the concatenation of the string derived from V1 to V7 in this order. And also we let GI be the prefix sum of these strings up to VI. So this is a kind of length, and we chunked this length at G1, position G1, G2, G3, and so on. So what we want to solve on this SG path, we focus on single SG path now. On this SG path is this problem, which we call the interval bias search problem. Given a position P and find variable Vi that contains the pth character in this time, pth character of U1 in this time. I skipped the details for this, but we showed that the problem can be solved in this amount of bits for this one SC path, which sums up to this amount of bits over all SC paths. Basically, this subproblem was also considered in the Beer et al. Paper, and they presented a slightly different data structure for this. But we simplified, we made some kind of simplification for this to make space smaller to work in this amount of bits. Next, I will show our SLP encoding version 1. We have two versions, actually. So, first of all, a variable, an internal node, is identified by an integer in from 1 to n, small n. Then we arrange the order of variables so that for any SCH, U to V, V is the next integer of U. This helps reduce space usage as we do not have to store the endpoints of SCHs explicitly. So basically, if we know that a child is on the same SC path, it is just +1 value. It has +1 value. So we do not have to store it explicitly. And the order of SC paths is arbitrary, which will be modified in version 2. Now, as an algorithmic tool, we use rank select data structures for bit arrays. Maybe this is familiar for the audience here, but I have one slide for this. For a bit array A of length small n, there is a data structure of n plus small of n bits to support rank select queries in constant time. And for B of 0 or 1 and position I, rank B AI returns the number of B's in A up to the position I. And given an integer k, select B AK returns the kth occurrence position of B in A. So in this example, rank 1 H6 is 2 because the is the number of 1 up to the position 6, which is 2. There are 2 occurrences of 1 up to the position 6. And the select 1 A2 is the second occurrence of 1 in A, which is 5. So this is rank select queries we use. So our SLP encoding version 1 stores 3 arrays. The first array, P, is a bit array of length small n that marks the last node of an SC path with 1. So suppose that this path, SC path, is the rth SC path. So the variables of this rth SC path have integers in this range. This path, the range of this path ends at the rth occurrence of 1, which is computed by select 1 PR, and starts at the next position of the select 1 PR minus 1. So these indexes are actually the integers given by these nodes. So next, if a variable is not the last node of an SC path, one of its child is on the same SC path, and the other child is the endpoint of the non-SC edge. And we store a bit array D of length n minus n prime to indicate the direction of the non-SC edge with 0 for left and 1 for right. In this figure, U1 corresponds to this position on D, and on this position on D, and its value is 0 because the direction on non-SC edge from U1 is left. And the value for U2 is this, is 1 because the direction is direction of non-SC edge from U2 is right. So this is the bit vector bit array D. The third array is the integer array E. E is an integer array to store the endpoints of non-SC edges. This V1 to V7 in this order. This is stored in this range starting from U1 plus R minus 1 to occupying 7 positions on this array. Now we can compute the children of a variable using rank select queries on P and D in constant time, following these arrows in this figure. So these arrows, the correspondence between arrays, can be retrieved by the rank select queries on these bit arrays. Now let's analyze the space usage of SLP encoding version 1. The bit array P with its rank select data structure uses n plus small o of n bits. And the bit array D with its rank select data structure uses n minus n prime plus small o of n bits of space. And since each element of the integer array E is stored in log n plus sigma bits, the array E uses n plus n prime times log n plus sigma bits. And adding the data structure for interval bias search on SC path, the total space usage is this. Next, we show our SLP encoding version 2, which changes this part, the array E, to store the endpoints of non-SC edges to work in this amount of bits, slightly reducing the, actually we want to delete this n prime times log n plus sigma bits of space from the space usage. And the version 2, SLP encoding version 2, achieves this. So our SLP encoding version 2. Now recall that there are n prime non-diff SC paths, and there are n plus n prime non-SC edges in total. So the n prime is the non-diff SC path. And these broken arrows, the number of broken arrows is n plus n prime. Now, for every non-diff SC path, we choose arbitrary one of the non-SC edges coming into the top node of the SC path. And consider a tree Q that has n prime nodes corresponding to the non-diff SC path connected by the chosen edges. In this figure, we choose these non-SC edges depicted by orange broken arrows coming into X4 from X3 and X5 from X3. For the SC path starting from X5, we can also choose the edge from X4, this edge, X4 to X5. But this choice is arbitrary. So in this case, we just choose these two non-SC edges coming into each coming into the top of SC path. The tree Q looks like this, where each node of Q corresponds to a non-diff SC path. The red and blue and green. So these are corresponding to these SC paths. Then we now arrange the order of the SC paths in some order on Q, say pre-order in this talk, so that the endpoints of chosen edges can be retrieved implicitly using Q. So in pre-order, this is the first node, and this is the second, this is the third. In the variables of the SC path is sorted in this order. 1, 2, 3. We can pre-process Q, the tree Q, to build a data structure of 2n prime plus or small of n prime bits to support the following queries in constant time. So navigation query on the Succinct tree. In a query, we are given the pre-order rank of a node U of Q and the integer K, and compute the pre-order rank of the kth child of U. And this can be done in this bits of amount of bits and constant time. Then our SLP encoding version 2 uses bit arrays P and D as version 1 did. But SC paths are now sorted in pre-order on Q. We also use a bit array M of length n plus n prime that marks the endpoints of the chosen non-SC edges by 1. So we have imaginary, we have the array E that was used in version 1 SLP encoding, which stores all endpoints of non-SC edges, V1 to V7. And we mark the position by 1 that correspond to the endpoint of the chosen edges. In this figure, we choose the non-SC edges pointing to V2, V4, and V6, for example. And then the corresponding positions on E are marked by 1, V2, V4, V6. Finally, we use an integer array E prime that is obtained by removing n prime 1 endpoints of the chosen non-SC edges from E. So basically, we specify the array E by removing the values marked by 1. So if mi is 0, the value ei is explicitly stored in E prime at the position computed by rank 0 queries on M. And if mi is 1, and it is the kth 1 in M, counted from this position on the array M, then ei is the top node of the rkth SC path, and which can be computed in constant time using the in constant time. So instead of the integer array E of version 1, version 2 uses E prime and M and Q, and the space adds up to this amount of bits. So this is the result, and let me conclude my talk. We proposed the new SLP encoding for all of Lagrangian time random access of this amount of bits. Note that the algorithm can be extended to extract L contiguous characters of T in all Lagrangian time plus the length of the contiguous characters we want to expand. So this is a list of future work. Firstly, we want to reduce the most space consumed term of small n times Lagrangian bits. And we want to extract L contiguous characters in this time, speeding up some leveraging word packed operation. Finally, we want to support random access from any variable x in time linear to log of the length of its derived string instead of Lagrangian time. So this is that's all. Thank you for listening.
Any questions or comments from audience? Oh.
Thanks, Tomohiro, for the talk. With this first piece of future work, is there a lower bound that you'll hit? Can you reduce this space-time without hitting UN Verben's lower bound?
This part?
No, reduce the most space consuming term.
This
Yeah.
and
Yeah.
Lagrangian.
Yeah.
I don't know.
But isn't there a lower bound preventing you from reducing it?
It's a
I guess
It's about the lower bound of time.
Okay, so I see if the time goes up, then yeah, okay, fine. And I had another question about supporting different queries, so maybe like rank or on the original string. Do you think you could adapt this framework to support?
Yeah, I think it's possible by adding some information on the...
Yeah, I think so too. Just prefix sums, right? Yeah, I think so. And also, this scheme seems very simple in some ways, so do you think it might be practical?
I think. I hope. But
Okay.
I haven't implemented it.
Yeah, okay. Someone else can implement. Okay, thanks.
Thanks.
Any other comment and question? Oh.
Okay. So it seems that it's not very cache efficient in that term that you have a lot of bit vectors where you need to do rank and select. So for thinking about practicality, if you do one access, I think you have to look up for each of the paths multiple integer arrays with rank select, right? So it's maybe not so practical, but it's also interesting for me. I mean, the time aspects, they kind of are a little bit in contrast to the space, maybe. I think you can afford, if you afford more space, then you can allow for extracting L-contiguous characters. But if you want to keep it in the same space, I think that's a challenge, right? What you want to see. Yeah. Because if you just blow up the space again, then it's easy.
Yeah.
Okay, thank you.
Any questions or comments? Okay, I have a small quick question, and I'm interested in your randomly updatable data structure. And is there any SLP encoding for supporting dynamic updates, or this method can be extendable to
dynamic operation?
Yeah.
I'm not sure, but it's not easy.
Let's discuss later.
Maybe for ending characters it seems possible, Like if you append characters, it seems possible if you change the bit vectors to be dynamic. But if you allow like insertion somewhere or modifying a character, it seems hard to me. But I just have seen the slides now, so I cannot give a detailed comment. But it seems possible for appending.
So let's thank the speaker again.
Thank you.
I'm Travis at Dalhousie University, and I'll be talking about how I've been having fun with MEMS in the past year or so, working with Nate Brown at Johns Hopkins, Laurie DePet at Ghent University, Mohsen Zakiri at Johns Hopkins, my and Nate's students Anas, Noor, Dov, Nitin, Suchit, and Hamza here at Dalhousie, and Ben Langmead at Johns Hopkins. I'm really sorry I couldn't be there in person, but I'll explain why during the talk. First, I'll explain exactly what MEMS are, maximal exact matches, and why we love them so much. Then I'll review Lee's forward-backward algorithm for finding MEMS, and a new algorithm called Boyer-Moore-Lee that I came up with last summer. After that, I'll talk a bit about why I'm more convinced now that bioinformatics matters than I was a year ago, and incidentally, why I can't leave Canada right now. Finally, I'll talk about a new approach to speeding up mem finding, k-mer-based breaking, or kebab, that I've been working on with Nate et al. So, first, maximal exact matches, or MEMS. A mem of a pattern P with respect to a text T is a substring of P that occurs in T but can't be extended in either direction and still occur in T. This means that if Pij is a mem, then either Pi is the first character of P, or Pi minus 1 to J doesn't occur in T, and either Pj is the last character of P, or Pi to J plus 1 doesn't occur in T. For example, the mems of MISSISSIPPI with respect to IPPISSISSIM are MISSISSI and IPPI. Notice that mems can overlap, but by definition can't nest. Some people call what I'm calling mems super mems or SMEMs, but ignore that for this talk. Finding mems, particularly long mems, is an important step in many bioinformatics tasks, such as taxonomic and metagenomic classification and DNA read alignment. In these tasks, we're trying to figure out where a DNA fragment comes from, either from which species or from which location in a genome. If the DNA fragment is actually close to something in our dataset, then even if it's noisy, there tend to be at least a few long noiseless stretches, and those tend to contain fairly long mems, which are unlikely to be the result of noise. For example, here I've generated a 550-character string over ACGT, shown below, chosen a 50-character substring, the line shown in red, made a copy of that substring, shown above, and replaced each character in the copied substring with probability 1/4. So the probability of a character actually changing was 3/16. The 9 characters that actually changed are shown in red. The numbers below the copied substring are the MEMS lengths at their starting positions. The 12 in parentheses means there's a mem of length 12 starting GATTCC, because I couldn't think of another way to write it. If we look at where the mems occur in T, we can see how long mems can help us figure out where DNA fragments come from. The occurrence of the 2 longest mems of lengths 8 and 12 are shown in red in the dataset, and they are indeed exactly where those mems were copied from. The occurrences of the rest of the mems are shown in black. The ones that look long are just 2 short occurrences overlapping. And while some of them are where their mems were copied from, most are just a distraction. It's hard to fit a convincing example on one slide, but trust me, people want to find long mems. Although there are algorithms with somewhat better worst-case guarantees, most practitioners use Lee's forward-backward algorithm, partly because it's practical, partly because it's incorporated into several popular tools he's written, such as Fermi BWT Mem and Rope BWT. Conceptually, forward-backward uses two FM indexes, which may be run-length compressed, one for T and one for T reversed. If you've forgotten how an FM index works, don't worry. All you need to know for this talk is that these two indexes let us find 1, the longest suffix of a pattern or of a substring of a pattern that occurs in T, matching character by character from right to left. 2, the longest prefix of a pattern or of a substring of a pattern that occurs in T, matching character by character from left to right. You can use the same two indexes to search for mems in the pattern's reverse complement as well, but I'm not getting into that here. Assuming every character in P occurs in T, the leftmost mem starts at the start of P. Since we can find the longest prefix of P that occurs in T with the FM index for T reversed, we can find that leftmost mem. By the definition of mems, the next character after that mem, written PE+1 here, has to be in the next mem. The start of that second mem, PS, is the start of the longest suffix of P1 to E+1 that occurs in T, which we can find with the FM index for T. Once we've found PS, we can ignore P1 to S-1 and, conceptually, recurse on PS to M to find the second mem and then all the other mems. Don't worry, there's an example coming up next. Going back to the same example, we start at the left end of the pattern, searching character by character from left to right for the longest prefix that occurs in the text. First we match A, then another A, then a G, and then a T. The next character, a C, doesn't match. That means the first mem is AAGT, and that C is in the second mem. To find the start of the second mem, we'll start at that C and match from right to left. First we match the C itself, then T, and then G. AGTC doesn't occur in T though, so the second mem starts with that GTC. Conceptually, we recurse on the suffix starting with that GTC, so let's get rid of the AA. Just in case, let's go through another rant. To find the second mem, we search character by character from left to right for the longest prefix of GTC, TT, ATA, CCC, AAA, etc. that occurs in T. First we match the G, then we match T, then C, then T, and then another T. GTC-TTA doesn't occur in T though, so the second mem is GTC-TT, and that A is in the third mem. To find the start of the third mem, we start it from that A and match character by character from right to left to find the longest suffix of GTC-TTA that occurs in T. First we match the A, and then T, then another T, then C, and then T, but then the G doesn't match, which we already knew from matching left to right. Anyway, that means the third mem starts TC, TTA, and we can recurse on the suffix of P with that G removed. But this is a lot of backing and forthing, so let's just summarize. We search from left to right and find the third mem is TC, TTA. Then we search from right to left and find the fourth mem starts CTTAT. And then from left to right and find the fourth mem is CTTATA, and so on. The operation to match a character or find it doesn't match with an FM index is called a backward step. The total number of these backward steps that forward-backward uses is proportional to the total length of all the mems, with the coefficient between 1 and 2 depending on the pattern of the text. This is quadratic in the length m of the pattern in the worst case, but usually something like m log n in practice, where n is the length of the text, with most of the total length coming from the many short mems, which are fairly useless. Can we find the long, interesting mems faster in practice by ignoring the short ones? Well, obviously yes, or I wouldn't be asking this, but how? Boyer-Moore may be the algorithm most famous for trying to ignore big chunks of its input. What happens if we mix it with Lee's forward-backward? Spoiler alert, this actually works. I called the hybrid algorithm Boyer-Moore-Lee, or BML for short. In Boyer-Moore pattern matching, we jump to the right the length of the pattern, then match from right to left until we find a mismatch or an occurrence. It's fast in practice because we often ignore a lot of the input. Can we use a similar idea for mem finding, assuming we're interested only in long mems of length at least L, say? Okay, I've told you we can, and it's not hard. Any mem of length at least L starting in P1 to L includes PL. We can start at PL and search from right to left to find the longest suffix PI to L of P1 to L that occurs in T. If I is greater than 1, then no mem of length at least L starts in P1 to I minus 1, because any such mem would have to include PL, and Pi minus 1 to L doesn't occur in T. So we can ignore P1 to I minus 1 and conceptually recurse on Pi to M. If the longest suffix of P1 to L that occurs in T is all of P1 to L, that is, if I equals 1, then we can fall back on forward-backward to find the first mem and the start of the second mem, then ignore everything to the left of the start of the second mem and recurse on the remainder of P. Let's set L equals 10 and go over the same example. It'll go faster this time, I promise. Going back to the same example, we start by searching for the first 10 characters of P, A, A, G, T, C, T, T, A, T, A, from right to left. We match A, T, A, T, and then T again, but then not C. Since C, T, T, A, T, A does not occur in T, we can ignore the characters up to and including that C, that is A, A, G, T, C. Now we keep going, but with the first 10 characters, the remainder of P. So now we're searching for T, T, A, T, A, C, 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 A, A from right to left. We match A again, C, C again, but not a third C. Since CCCAA does not occur in T, we ignore the prefix TTATAC. Again, we're looking for the first 10 characters of the remainder of P. We match C, A, T, T again, but not C, so we ignore the prefix CCAAAC ending with that C. We're actually moving pretty quickly. Now we search for TTACGGATTC from right to left, and match C, T, T again, A, and G, but not another G, so we ignore the prefix TTACG. Now things get a bit more interesting. The next 10 characters do occur in T. In fact, they're the start of a mem of length 12. We don't know that, of course, so we just search for the next 10 characters from right to left. T, T again, G, G again, C, C again, T, T again, A, and G. Since we've matched all 10 characters, we fall back on forward-backward. We find the longest prefix of the remainder of P that occurs in T matching from left to right. G, A, T, T again, C, C again, G, G again, T, and T again. Actually, we already knew that GATTCCGGTT occurs in T because we found it, searching from right to left. But unless we're using a fancy bidirectional FM index, we have to start over from scratch to switch from searching right to left to searching left to right. We match another T and a G, but not the next character T, so GATTCCGGTTTG is a mem of length 12, which is at least L equals 10. To find the start of the next mem, we follow forward backward and search from right to left, starting with that T that didn't match. T, G, T, T again, T yet again, G, but not another G, so the next mem starts GTTTGT, and we can ignore the prefix GATTCCG. And we go back to looking for prefixes of 10 characters, searching from right to left. Nothing really interesting happens. We find a mismatch, we ignore a prefix. We find another mismatch, we ignore another prefix. Until we find that the last 6 characters of P do not occur in T. Normally, we'd ignore the prefix GAAT and search for the next characters in the remainder of P. But there aren't 10 characters in the remainder of P, only 5. So there are no more MEMS of length at least L equals 10, and we're done. BML is still slow in the worst case. To see why, suppose we're looking for MEMS of length at least L equals M over 2, and every L minus 1 mer in P occurs in T, but no L mer does. Then for each L mer, of which there are M over 2 plus 1, we take L minus 1 backward steps, so we take theta M squared backward steps in total, but then we don't report anything. Nevertheless, BML still seems faster in practice than forward-backward, so I emailed Heng Li, who designed forward-backward and lots of other things. Thinking about this months later, I realized how lucky I am to be a professor. I'd spent quite a long time thinking about mem finding, but when I'd finally found something I thought was really cool, I could just send it to the person I thought might make best use of it without asking anyone's permission. I don't know if you get that kind of freedom in industry. So, who is Heng Li? Well, he's an associate professor of biomedical informatics at the Dana-Farber Cancer Institute and Harvard Medical School. He has over 250,000 citations on Google Scholar, many for papers on which he's the first author, including those introducing the SAM format, the first paper in the list here, BWA, the second and fourth papers, and BWA MEM, the fifth paper. Please remember that he's at the Dana-Farber Cancer Institute as well as Harvard. It'll come up again later. Here's my email sketching BML in one paragraph sent on Saturday, July 13th last year. Here's Heng's reply sent on Monday, July 15th, 2 days later, saying he'd implemented BML in his tool wrote BWT3 and sometimes got over 10-fold speedups, all in a weekend. When wrote BWT3 was published, I was in the acknowledgements. As you can probably tell, I'm quite proud of this. Please note again that Heng is at the Dana-Farber Cancer Institute as well as Harvard. So, why do I care so much about the Dana-Farber Cancer Institute? To explain, I'm going to have to pause for what I think people in computer science call a war story. On October 11th last year, I was diagnosed with blood cancer, either lymphoma or leukemia. The next day, that diagnosis was refined to B-cell acute lymphoblastic leukemia, or BALL for short. That refinement cheered me up a bit, and I really needed it just then, because BALL isn't the most lethal blood cancer. B-all is crude British slang for nothing, as in, get back to work, there's B-all wrong with you. To treat BALL, the doctors said they were putting me on the Dana-Farber chemotherapy protocol, version 4, I think. So of course I said, I know someone there, which was exaggerating slightly. But when I told that to one of the doctors, Dr. Trache, it turned out she knew about BWA and bowtie. So real doctors actually do care about bioinformatics. Of course, when Ben came to Dalhousie to give a talk, I made sure I got a picture of them together. Dr. Trache is an expert on heritable blood cancers and a real doctor who sticks people with real needles. For example, this is the needle she used to give me a bone marrow biopsy. As you can see, it's nearly as long as my hand and thick enough to drive through the bone to the marrow in my femur. I consider that a very real needle. Fortunately, it hurt a lot less than you'd think. Even more fortunately, that biopsy showed that chemotherapy had got me into remission. Yay, Dana-Farber. Unfortunately, another biopsy with another such needle a month or so later showed that chemo wasn't wiping out my leukemia entirely, so without another treatment, I would probably relapse. Of course, I asked Google what my life expectancy was with drug-resistant ALL, and it said, not much. For adults with drug-resistant ALL, median overall survival is often less than 6 months. Fortunately, again, the hematologist in charge of my file, Dr. Marcoux, told me Google's out of date. That was true 5 or 10 years ago, but we have lots of new therapies now. I'd say your chances of surviving 5 years are 60 to 70%, and after that, relapse is rare. I ended up getting a bone marrow transplant in May, which is why I can't travel for a while. Sorry again that I can't be there in person. Anyway, biomedical research, probably including bioinformatics, is actually really making a difference. So without further ado, let's get back to mem finding. When I got sick, I was teaching a course on compact data structures in bioinformatics to 5 undergrads, Anas, Noor, Dov, Succinct, and Hamza, and 1 grad student, Nitin. In the first month, we got through FM indexes and R indexes, the move structure, and forward-backward, and I think we were just getting into BML. The dropout rate was quite high. Those were the 6 who actually stuck with me. When I went into hospital, I got my former master's student, Nate, who's now doing a PhD with Ben, to take over the course, backed by Ben and his postdoc, Mohsen. I sketched an idea for speeding up mem finding using a k-mer filtration pre-processing step, and Nate et al. Turned it into an assignment, and then a course project, and we just turned that into a Spire submission. We call this idea k-mer-based breaking, or kebab for short. Let's start with some easy observations. First, a mem cannot contain a k-mer or any substring that does not occur in T. Therefore, the maximal substrings of P of length at least L greater than or equal to K, consisting only of k-mers occurring in T, contain all mems of length at least L. To see why, consider that any other substring of P is either a substring of one of those maximal substrings, or it contains a k-mer not in T, or it has length less than L. A Bloom filter can return false positives but not false negatives. I'm not going into how Bloom filters work, but trust me on this. Therefore, the maximal substrings of P of length at least L greater than or equal to K, consisting only of k-mers occurring in T, according to a Bloom filter for the distinct k-mers in T, also contain all mems of length at least L. It's easy to find these maximal substrings with the filter, which we call pseudomems, because they're our best guess at the mems. Well, we call them pseudomems because they're our best guess at the mems given only the information in the filter. If T is highly repetitive, such as a database of many human genomes, then the number of distinct k-mers is small compared to the length of T, for reasonably small k. This means the Bloom filter can be very small compared to T and still return false positives with only low probability. We're interested in highly repetitive datasets because it's better to align DNA reads against many human genomes than against only one, because it helps prevent reference bias. Using pangenomes is also better for classification. Ben has whole invited talks on YouTube about why reference bias is bad, so I'm not going into it here. Computing k-mers hashes and checking the Bloom filter is pretty fast compared to backward stepping because of locality. If L and K aren't too small, then the total length of the pseudomems is often much less than the length of P, especially if P is a bit noisy, a long read, for example. This means finding the pseudomems and then searching in them for long mems is often much faster than searching for mems in all of P directly. At first glance, this example looks like the previous ones, and the pattern is the same, but the dataset is different. Now pretend we're seeing only an aligned part of a dataset of 11 genomes, the part the pattern should align to. I generated each of the 11 aligned substrings in the dataset from the original substring of the dataset, the one I copied the pattern from, by replacing each character with probability 1 over 10. The maximal substrings of P consisting only of 4 mers occurring in this part of T are shown in black below P itself. Why only in this part of T? Because I was trying to fit things on one slide. This is just a toy example. For k equals 4 and L equals 10, GGATTCC GGTTT is the only pseudomem, and GATTCC GGTTTG is the only mem of length at least L. You can see why, for good choices of K and L, focusing on pseudomems speeds things up. But enough of this particular unconvincing toy example. Here's a better toy example Mohsen did for the paper to show how everything fits together. If you look closely, the reference AGCCCTAGCCCTAGGCCCCG is fairly repetitive, and it contains only 12 distinct k-mers instead of 16. Given a pattern, we check its k-mers against the filter. Now we're using 5 mers in this example. Find the pseudomems and then pass them to a normal mem finder. If we want only the t longest mems, then we can search the pseudo-mems in non-decreasing order by length and stop non-increasing order by length, and stop when we have found t mems at least as long as the next pseudo-mem. But really, does all this really work? Nate reran the metagenomic classification experiment from Ahmed et al.'s Spumoni 2 paper from Ben's lab, published in Genome Biology, with a data set of about 6,000 genomes from 7 microbial species totaling about 27 gigabytes, a set of about 10,000 reads from yeast, which was not one of those 7 species, about 20,000 bases long on average. Those are the null reads. A set of about 600,000 reads from those 7 microbial species, about 25,000 bases long on average. Those are the positive reads. The top graph shows the mem finding time overall for k equals 20 and various values of L. Running just rope BW3 with BML is the blue line. Running kebab followed by rope BWT3 with BML is the orange line. For the null reads, which don't have many 20-mers in common with the dataset, just running kebab first made a huge difference for L equals 40. For the positive reads, running kebab first roughly halved the total time, again for L equals 40. We didn't want to modify rope BWT3 to take a parameter t and stop early after finding t mems as long as the next pseudomem, but we estimated how long it would take by working out where it would stop and then giving it only the pseudomems it would actually search. Our estimate is it should be really, really fast. To see how downstream accuracy is affected if we use only the longest 10 mems instead of all the mems of length at least L, Laurie and Nate reran the metagenomic classification experiment from Laurie et al.'s Tagger paper, just published in RIKEN-AIP-Day. This used a dataset of about 8,000 genomes from 8 microbial species, totaling about 37 gigabytes. Laurie suggested that instead of searching pseudomems until we find the longest t mems, we just take the mems of length at least L from the longest t pseudomems. This is a hack. It's not guaranteed to find the t longest mems, but it seems to work. The graph on the top left shows Tagger's accuracy using the longest t pseudomems, the longest t mems, and all the mems of length at least L. In all cases, we used L equals 25 because that was found to work well in the original Tagger paper, and k equals 20. Notice that for t at least about 10, using the longest t pseudomems doesn't seem to hurt classification accuracy, and it significantly decreases the number of backward steps, that's the chart on the right, and the overall time, the chart on the bottom. To finish up this talk, it wasn't in the paper. I asked Nate to rerun the Spumoni 2 experiment using rope BWT3 without BML, with just forward-backward, rope BWT3 with BML, kebab, and then rope BWT3 with BML, and kebab to find the 10 longest pseudomems, and then rope BWT3 with BML on those pseudomems. It would take more experiments to fully back this up, but basically, this graph shows that in less than a year, we've seen something like a 40-fold speed up in practical mem finding with pretty simple techniques. Thanks for your attention, and please feel free to email me any questions.